microphones off and your pictures off. What we need you to do is raise your virtual hand. So again, at the bottom of the screen, there is a box that says participants. If you click on that, it will open up the participants box. And at the bottom of that box, it will say raise hand. If you raise your hand, you'll come at the top of our list and we'll see in order, and we'll kindly ask you to then unmute yourself. And please, it is better if you can share your video. You don't have to, but it is good if you do. And then everyone will be able to see you, and you can pass direct comments as well as what's going on inside the chat at the same time. It is a community discussion, as Derek has already said. Okay, so we're not here to pronounce anything or any decisions. We're just here to give you the best options and hopefully see what practical approaches people are having. One other point of order is this meeting is being recorded, and unlike the previous two, we do plan to share this at the end. So please also bear that in mind, no swearing. And finally, there are no planned fire alarms, and I hope you all know where your nearest toilets are. At that point, I shall hand over to Rachel. Thank you.
Hi everybody, I'm Rachel Walker. I run the Flowcytometry Facility at Bagram Institute in Cambridge. So just to go through the topics we're going to discuss, as Pete's already mentioned, we're going to do about half an hour on each topic, but some of these topics will overlap. So if we're talking about one topic and we find that the discussion's moving into something we're going to discuss later, we're just going to move that discussion on to a future point. So we're going to start by talking about cleaning. So this is cleaning of equipment within the lab. Then we'll talk about PPE, and then physically distancing, and then we'll take a short break. Judging by the other ones of these meetings, that'll probably be about 3:45. It depends on how our chat goes. And then after our break, it will be talking about managing staff, and then staff user interactions and training. Can I also ask everybody, quite a few people have already done it, but if you can make sure your full name is down as the participants, because if you want to ask a question, it's easier for us to call you out by your full name than we've had some that have just come up as numbers. So thank you, if you could change that.
Yes, exactly. Otherwise, we'll be calling it Z342 or whatever Elon Musk's child is called. Okay, so we're going to start off by thinking about cleaning. So the slide we've put up here, I'm not going to go through all of those points particularly, but these are some of the things that we might want to consider within the lab. You know, things are going to be changing, aren't they, when we go back to the lab? This will come out multiple times during this afternoon. We're going to have to work in different ways. So one of the things we want to consider is how that we make sure that everything we're going to use in the lab is clean and fit for purpose. One of those things may be, whose responsibility is it? Is it going to be with the user? Is it going to be with the staff of the facility? And because we're flow-based, is there a difference between analyzers and sorters? If we're going to clean our machines, how are we actually going to do that? Lots of different ways out there. So we want to hear from you, really, about this. Are those of you who are back at work already? Have you changed the way you're cleaning your equipment? Those of you who are going back, have you thought about this already? Is there something in place that's going to enable you to do that? We wait for some raised hands now. Otherwise, Peter and Rachel and I will talk for hours.
We have no problems with that front. It was interesting. We did draw up a quick list of different things that possibly need to be cleaned in the lab, but I'm sure there's a lot more points, and how we actually go about cleaning those would be interesting to get an idea of what people are actually doing at the moment, if they are already back in the lab, or what they're planning to clean or not clean, and who's going to clean it as we go through. So it'd be good to get some different opinions. At York, we're still undecided. We're debating each of these points quite carefully as to what to do, and actually this is going to help us decide what the best practice will be, because we are not yet convinced of any solution, even after 2 days of meetings.
Excellent. Okay, so Carol-Ann, do you want to say something? Jump in, unmute yourself.
Hi, can you hear me?
Yeah.
Yes.
Yeah. Shall I put my video on as well?
Why not?
I can't seem to get it to do that. Every time I try to click on the video, it just won't do it. Sorry. I was just going to say I'd been thinking about cleaning, Oh, start my video. Hi. I've been thinking about cleaning for when we start back, and what I was planning to do was for the technicians in the lab to wipe down all the surfaces, keyboards, mouses, and all the sort of high touch points before anybody comes in. So sort of as a part of setting up the lab in the morning. And then I was going to get the users to then clean down all the bits that they're going to touch before and after they use the instrument, so that the sort of responsibilities shared in terms of the technicians do it all thoroughly in the morning, but then each user then re-cleans it when they come in, because they're then, so they know they're happy to touch it, and then they clean it all after. But then when the next person comes in, they can wipe it all down again, because then that makes them feel more secure that it's been done properly. So that was my plan. I don't know what everybody else thinks of that.
Can I make a comment, Derek?
Yes.
Yeah, Caroline, I think that sounds very similar to what we were thinking at the moment. I've noticed that someone has pointed out the use of 70% ethanol as a spray. I don't know if anyone's got any thoughts on using ethanol sprays over just a sort of a wash bottle to actually put onto a microfiber cloth or similar, or Kim wipes.
Well, I think we use something called BioCleanse in the lab. So we were going to just have spray bottles of that. Because we've been told in the past, actually, that you're not supposed to use ethanol for cleaning because it breaches the license that you have for actually buying ethanol. But I don't know whether that is now.
I've not heard that one.
I think we would prefer not to use a spray if we can avoid it.
I think Karen Hogg says prefer. I think you should say prefer not to use a spray, because it does have health implications. And someone pointed out recently that there's also, because there's a lot of electronics around, you shouldn't be using sprays that can generate aerosols from it. So possibly a bottle may be better.
Somebody's asked about using Virkon. I've always been a bit worried about Virkon and metals. We've had problems with some of our tissue culture hoods going rusty, even though they should be stainless steel. So I don't know what people's thoughts are about putting Virkon, whether or not that may be a bit harsh if everybody's cleaning the tables and the machine, whether or not it might cause more damage.
Yeah, so Andy Filby's come out with a good comment as well. Guys, you can actually put your hands up and actually say some of this. You don't have to type it all as big essays.
No,
We do welcome
it's a lot quicker to actually say it, I think, than type it.
I can't even see Andy's comments now.
Can I just ask, I can't seem to unswitch off my video camera. I don't know if you guys can do that from your end. Thank you.
I think we can, yes.
Yeah, there you go. As Andy said, that's a very key point. A common question actually over the last few days: is 70% ethanol proven to kill the virus on contact, and how long does it need?
Okay, we've got a couple of hands. Bangwen from GSK. Do you want to jump in, Bangwen?
Yeah, hi, Derek, and thank you, and happy birthday to Peter.
No, thank you.
I'm just want to say that we had a general discussion within GSK when we had the Q&A session with the EHS officers. So the discussion is that there's various methods that are available to kill the bacteria and virus like SARS-CoV-19. But then the general understanding is that the limitation with the use of the bleach or Vicon for surface cleaning is that they are corrosive and not suitable for metal surface, as our member already mentioned. So the additional risk is that with the chlorine gas generated is cancerogenic. So over here, that's why in GSK, at least we agreed to using ethanol to just wipe. If we can use that to clean thoroughly, that would be helpful for us. So I think most worry is actually when you have a block in the in the lines, and you will have any errors generation. But in that case, that most of our work, that's what I see the risk assessment is, we will have the cell sorters inside the biosafety cabinet hood, which is class 2. And if it is not possible over there, then you need to have a proper risk assessment in place to see that where you can inactivate your samples. And there's already various guidance on how you inactivated the cells. And I guess that I would like to hear people's opinion about how to do that.
I see Andy's put his hand down now. Andy, did you want to speak?
Yeah, why not? Okay,
Why not, indeed?
So I was just going to expand upon my comment, really. I mean, you know, so we're working with COVID samples, and we have done quite a thorough sort of risk assessment of, you know, how to clean afterwards and keep, you know, the contamination very much eliminated from our working areas. And there are a number of papers from, you know, probably the last 10 years where there's obviously the previous SARS viruses as well, and MERS. And ethanol does seem to be 70% ethanol does seem to be extremely effective. And there's a paper showing that 2 minutes is enough to deactivate and kill the virus. And then I think Virkon at 10%, and then you get into the formaldehydes and stuff like this. So this is a challenge because we typically use a microsol spray as our SOP to clean, but we had to contact the producers of microsol to find out if it is actually they've got any data, because that's the problem. You can either go two ways. You either have to prove it empirically or rely on a reference where it's been proven empirically. So that's all I wanted to say. Really, it's a real challenge.
Yeah, I think there are a few papers, as Andy says, that have shown the effectiveness of alcohol, 70% alcohol there.
Yes, I've certainly done some research and found just that 70% ethanol is very effective. And of course, doesn't leave any residual, any residues actually on the surface as well. It's a much cleaner way and optically better for other components as well.
So has anybody also considered maybe the use of those wipe clean keyboards as well? So, I mean, the cleaning's still an issue, but it may be easier to clean them. So Heather, you have a comment?
Can you hear me?
Yes.
Yeah.
Okay. My facility is worried about using sprays because they have a feeling that it may re-aerosolize any viruses that have settled down on the surface, which I think is a bit strange. And they also suggest cling filming communal equipment, keyboards and mice, and then swapping out the cling film between each users. I don't know whether you think that's a good idea.
Yeah, I think this is something that came up in the light microscopy one as well. Pete might say more about this, but yeah, the idea of cling filming everything that somebody's likely to touch between there, which obviously means a lot of cling film.
Yeah, and it may be a way of saving. So I know some people are considering wearing gloves, and we'll come to that in a moment as well for the PPE side. But yes, it can save on that. It's just putting cling film loosely over the top of your keyboard has been found to be quite effective. Was recommended very early with the bioimaging community, even to the point of putting it around the eye cups of their microscopes, any contact points of trying to shield it. But it also protects them, so you can actually spray and clean those. So you can leave some of the cling film on and clean between users if they wanted to as well, more effectively.
Absolutely, but Anne makes a very good point that it's not good for the environment, and it potentially is a lot of waste that we've got to get rid of as well, when we may not have as many people removing our waste.
There is on the email invitation, you should have all had 3 different links as well. So from the UK government, we have now guidelines of how to best operate in a scientific lab. It's one of the very few places they have actually given a prescription of how to go back to work, and we are one of those. And they're quite prescriptive of not to do more than what our normal routines are. A lot of social distancing part of it more than anything else. So that'll be well. I'll find the link and I'll put it onto the chat in just a minute.
Yeah, and Katie makes a point there that there are keyboard covers available as well. I think I was looking those up, in fact, the other day. So again, we can share links to these sort of things that are potentially useful when we get back. But I'd just like to go back to the idea of who's actually responsible for this, because as we'll see, our working practices are going to be slightly different when we go back. We may not have staff there the whole time, so we're going to have to trust our users to do this. So if anybody's got any comments on that, that would be useful to hear, I think. Karen, though, you have your hand raised.
So can you hear me? Yep.
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 I think in terms of who cleans the instrument, touch surfaces, keyboard and bench around it is going to be quite site specific, because some people might only have one person doing everything and some people might have a team. But even with a team, monitoring and cleaning after every user is going to be quite labor intensive. So I think I would feel that the users need to take some responsibility for wiping down the surfaces, keyboards and things they've touched themselves with whatever we decide is appropriate to do so with. Maybe the staff, be it departmental or core technicians, go in, do it maybe at the beginning and the end of the day. So I think it would be a bit onerous for us to do it all. So
no, I think you're right, Karen, and I think you also bring up the point that we cannot be there all the time for the users. We have multiple systems, and we can't be sure that we would get in before another user. So I do think they'll have to take responsibility for themselves personally.
It'd also be more people going in and out of the rooms if we can keep having to go in at the beginning and end of every user's session. More times we touch the door handles and the components within the room. So I think that needs to be considered as well. It's nice, we're very keen to do things for people, but we need to make sure we're not making the situation worse by trying to make it better.
Absolutely. Alfonso, no, sorry.
Yeah, Caroline made that similar point at the start, that she was, I think, planning to clean the room at the start and end, but users have responsibility during that time.
Yeah. Alfonso, I'll bring you in a second. I just want to address a point that Andy Herman had made in the chat about do people have a checklist to make sure that people have cleaned? We certainly do in terms of the machines at the moment, but I don't think anybody's got one in terms of cleaning around there. That may be something we all have to introduce. Now, Alfonso, can I bring you in?
For me, it will be mandatory to clean the area around, and that will be to make sure that we'll have a 1 hour gap, not 30 minutes, 1 hour, because I guess there will be some queues as well upstairs with centrifuge and tissue culture labs, etc. So to avoid that you have any kind of compromise to have 2 or 3 people waiting to be entering and keeping them on the door, we introduce 1 hour. 30 minutes for making sure that they clean everything correctly, and then half an hour to ventilate as well the air in the area.
Okay, there's another point in the chat there about making sure that people are sanitizing before entering and leaving. That may be something that we may have to think about. Not every room has that facility in there at the moment.
Of course, a lot of these things will also be dependent on our local institute and what guidelines that they dictate down to us. We're going to have to work within that framework. We can only really add extra layers if we think necessary, and we will probably have to justify that. We can't just suddenly dictate that people should go above and beyond what they're having to do in their own lab without very good justification. That we will have to, which is hopefully, if we can get some consensuses here, if we think something is more, this gives very good ammunition to go back and say, well, look, actually, because this is a shared resource, people coming in from all sorts of places, they're working in their local teams to minimize spread, but then all these teams are mixing in our facility.
Yeah, it's an important point. In the core facilities are not the same as the research lab, where everything is probably slightly more regimented and people know where they're going. There will be a lot more mingling.
Do you think there's a danger that some people may clean before as well? So therefore you're ending up with people, would people necessarily trust the person before them to have cleaned, and therefore they're going to clean? So should perhaps the person who goes on the machine clean? So the core cleans it as suggested first thing in the morning and lunchtime, but we ask users to make sure it's clean before they start.
Andy?
Oh, no. Alfonso, you're back there. Do you want to say something?
Back to that question, comment. Again, that's for me interesting to put the 30 minutes on top of the other 30 minutes, because if you don't trust the others, I will clean it before I start. So I want to make sure I clean and then I will clean.
Vai fazer xixi antes da aula da professora, se faz favor?
I will basically don't trust anyone nowadays. So I will clean myself the area, and then as soon as I finish, I will clean it as well.
Exactly. I think that's what's likely to happen. Andy Riddell, you want to say something? Are you there, Andy?
See it?
I can hear you.
All right. Okay. Here's the video. Is that
There you go.
If you're going to clean keyboards and stuff in between everything, there's a big burden that's placed on the staff to do that, as we've kind of discussed. If everybody's given the equipment to clean and they clean before and after, then that's fine as well. But the thing is, I mean, this is really probably the minimum point where you're going to get infection from your staff. Actually, what's missing in this is actually them traveling in is going to be the biggest vulnerability, especially on public transport and things like that. So no matter where the staff come from, they're going to bring in whatever they bring in within the environment.
Yeah.
So clean getting people to clean before and after themselves seems to be a sensible idea.
Yeah. Yeah. So we put these points up, but then they're not standalone, are they? There is going to be a big overlap between this and the PPE, and indeed we will talk about getting people to work and how they're going to work.
Has there been anything out from any manufacturers on how they recommend we clean? Have any of the manufacturers said anything about their best practices that they recommend?
I'm pretty sure some of them have. I'm not sure I've got the right notes. Actually, Telma, hang on a second, because I know Bangwen has done something about this. Bangwen, you want to say something?
Yeah, so I already reached out to the major manufacturers like BD, like Beckman Coulter and Mutiny, because these are the major cytometers we have in GSK. So a lot of them really just referred us to talking back to the EHS officers or looking at WHO's recommendations. So they don't have a clear guidance on like what you must clean with. And for instance, with Mutiny, they also say most of their machines are very small, so you can put it in a biosafety cabinet. And over there, then, especially if it is ducted vending, so not the recirculating cabinet, then you are safe to operate inside. And we also get some references that's about the aerosol generations. But again, it's a focus on how to safely minimize it. It's a guidance. It's not really saying that you must do or you not do.
Okay, thanks. Telma, did you have your hand up? Or did somebody lower it?
Exactly. I just wanted to comment because we are already working, so our facility never really closed. During this time, we just diminished all the work, also to the institute itself. But I can start my video. Yeah, sorry. Can I? Yeah. Hi.
There you are.
And hi guys.
Hi.
And what we are doing is basically like everybody cleaning everything, but we are using actually the swipes. So I think that's also very practical. So you're not spraying anything. And the ones that we are using, I think it's also used at the hospital, so they are very known already. And it's actually very efficient. They also are very humid, so you see that everything stays wet for a while. And we are saying the users before and after, like you were saying. And but we don't have the limit, like Alfonso was saying. So we have 3 instruments in one room. We blocked one because of the physical distance that we can discuss later. But anyway, we don't have the limit of waiting some time between each user. Everybody has to wear a mask at all times, gloves also, but we don't have the limit of time. But yeah. Let's see. Right, it's always a difficult time, and let's see how it goes.
Yeah, thanks, Selma. So how are we doing for time in this section?
Okay. It's interesting. We have had a few that are using bleach as well as ethanol. In the microscopy community, we heard a lot about sodium using alkali as well as some detergent and an ethanol mix, hybrid of it. So it'd be interesting to know from those who think that bleach is having some bleach present is better than just 70% ethanol, and why, what is the best solution to this? We'll have a vote actually in a little while about is 70% ethanol enough, or should we be using more, such as the ethanols used, such as bleach. So if anyone's got any comments on that, it'd be really good to hear why that's been encouraged, because I've certainly seen arguments on that on the listserv for using hybrid solutions. Gosh, I know it's Friday afternoon, guys, but come on.
Are people doing anything different for machines that their staff are running? So sorters and maybe any of the benches with the samples? Are people doing any different, or are you still going to use the same cleaning as you would with the analyzers?
We'll be using the same cleaning. Actually, how are we going to transfer samples? That's usually a user passing the sample to us. When we're doing a sort for them. We'll come to how we interact with the user when we're optimizing it for them in a little moment's time in the third session. But how are we going to actually have samples passed from users to us?
Okay, well, Chris, you want to say something?
Chris Allen?
You coming on board?
Yeah.
Oh, there we go.
Yes.
You're there.
So at Colorado State University, the university has gone into production of their own sanitizer. That sanitizer is 70% ethanol, but also has a percentage of hydrogen peroxide and a little glycerol. Now, I think that this is mostly like a hand sanitizer formula, but we have produced this stuff as a university. That solution has been tested here at Colorado State. We have BSL-3 labs, quite a robust infectious disease program, and so it has been shown to be effective against COVID-19. So you ask about other formulations. I don't have the particular formula for the Colorado State sanitization solution, but I can sure dig it up and share it with the group if you'd like. That's all.
There's a lot of comments coming in on the chat about the samples in the sorter, but I think maybe that's something that we will cover a little bit later on.
Yeah, so
where are all my hands gone?
Yeah, so Bangman's just come back in with the comment of the danger of having any bleach present for that rusting and corrosive nature for different materials, certainly with metal surfaces, for example.
Okay, Karen.
So when people bring their samples into the sorter room, usually they have handled those samples with gloves on anyway when they've been in their lab. So it shouldn't have general touch surface contaminants on it, hopefully, because they would have been handling their sample either in a tissue culture hood or on the lab bench with gloves on. And at that point, that's mainly to protect the sample from any contaminants that they're going to put in the sample. So the amount of contamination would be or should be very low. And if people have got gloves on, they're less likely to touch their face or touch other surfaces that they shouldn't be. So I think if you having samples passed to you from a user, you'd probably have gloves on as well. I certainly would before I put them on the sorter, and then I would put them on the sorter and I would be then not consciously touching my face.
Yeah, I think, yeah, as a biochemist, the gloves are to protect us as well, not just protect the sample, because very much you don't want the chemicals going near you. So as soon as I've got gloves on, I would not touch anywhere near my face, because that means something's there that shouldn't go near me. So I think actually having gloves on is very good at preventing touching, because I'm constantly touching myself. That sounds so wrong. I've got to be constantly mouth, nose, sitting here. And if I had gloves on, there'd be no ways I'd go near that. I think Lucia came out with a comment. If they're always wearing gloves, where's the protection?
Yeah, I think that there is that to it, but while you've got the gloves on, you only have the gloves on for a short period of time. So I think the risk of there being something on the outside of a sample tube is lower than it being on their hands generally. That's the point I'm trying to make.
Yeah, I think we're slightly sneaking into the next topic as well, which is PPE. So actually, Bangwen wanted to say something, and then maybe we'll go to the poll and move on to the next section, because I think we're about ready for that. Bangwen,
Yeah,
you come back in again?
Sure, I'm totally agree with what Peter said, that if you have some gloves on, this will be helpful. But I just want to actually revisit this cleaning sample bit that we talked about. The ethanol cleaning will be less corrosive to metal surface. But after checking actually the guidance in our disinfectant for laboratory use is saying that we're using distill to do the cleaning over the ethanol cleaning. The reason is that it's non-toxic and may decrease the skin on a prolonged contact. While when using the ethanol, then it has a poor penetration to organic matters and should be used only on the physical cleaning surface. So that's why we choose the distill over the 70% ethanol in the inactivation or disinfection when we do the cleaning.
Is everyone familiar with what distill is? So I know some over in America at some of the other meetings weren't that familiar with it. If anyone has a link that they could put onto the chat, so some say yes, some say no. So not everyone's actually
Distill's contrad, isn't it? Isn't distill and contrad the same thing?
No, the contract
so.
is that you cleaning the machine. That's a distill is the, like you can spray on the on the paper and then you clean and wipe.
Yeah, sorry,
and I think there's alternatives called Tego 2000 or Microso. I can type those in.
Yeah, if you could put those links in there, that would be fantastic.
I think George has put there, yeah, contrad is what we used to as decon in the UK.
No, it's different, yeah.
Yeah, thank you. Really
yeah, we do have links there, so we can put those up as well. So we should sort of move on from this section. As you can see, I've just put up a couple of polls. We need to open that poll, which, oh, there we go. That's open. So it's fairly simple. Just put in your response. All anonymized. Once we've got a reasonable number, we'll stop that.
Rafa, thank you very much for putting that link in. Hopefully most can see the poll.
Yeah, it'll disappear once you've voted. But once we've got most of the votes in, we'll stop that, and then we should be able to display the answers to that. We get to know Zoom a lot better at the end of these things, don't we?
Get to know us a lot better as well.
Yeah.
Especially the second session, you've had your wine, Derek. I think that's drawing to a close.
Yeah, that's near enough there. Should we stop that?
Yeah, share results.
Okay, so hopefully everybody can see that. In terms of responsibility clean, most of us seem to think that each user should be responsible for that. But of course, it may depend, as we've discussed, may depend on the time of the day, how many people are available to do that. And it looks like most people are fairly happy with ethanol. As we said, there are a couple of papers. Again, we'll put the links if they haven't already been put in to the chat. Okay, so we should probably move on to our second topic.
Yeah.
Which is PPE.
I'll pick this up, Derek, for you. I've just closed the poll. So a good question. We've already started talking about cling film wrapping, but actually what should users be wearing in labs? And I know this is quite some polarized views over the last couple of days of whether people should wear gloves, should not wear gloves. How easy is it for people to wash their hands? There's not always sinks in every lab easily available for them to use it. So these are some of the points we've thought of. So actually, if anyone's got any thoughts or questions or comments, it'd be great to get your hands up or get going in the chat box going on. We do have to consider what we use is less, there are a strain on some of these resources still. I'm aware that latex gloves are limited still. We can buy them. They're not only for NHS services, there are more than enough there, but a lot of the labs did donate a lot of gloves into the NHS and to their national health services around the world, which means when they start back, there's going to be a big glut wanting them, which actually means we may actually not have that many accessible to us. Someone beautifully pointed me, I was quite keen to have gloves ready in the lab. They always do for handling samples. I think we need them to handle most of our samples anyway, but they could be pinched quite quickly as other labs have a lack of such equipment. So what's the best way to use that? It's a nice point there about masks, whether we should wear face masks. I don't know what people's opinions are on that. Cameron Hogg, quite clear guidance actually from in the UK at least, is that we should not necessarily be wearing face masks unless we ordinarily have to. But it is something that is optional. It's the only PPE that they say is optional. Everything else should only be through what the science has done. Chris,
yeah.
Hi, it is quite interesting that the UK government don't advise using face masks. But it very much depends on where you are, because in Italy here, it's by law I have to wear a face mask indoors when I'm surrounded by other people. So we have to use gloves, face masks, and we also have to keep 1.5 meters distance from each other. I think it very much depends on your local laws as well as what you think might be right. Whether the laws are right is debatable, but you have to follow them.
Chris, thanks very much. And absolutely right. We do have to follow local rules. Lucia, I see you asking about training, and we'll come to that with the next few topics about physical distancing and staff user interactions, where, as Chris said, there may be instances where extra PPE may be required to enable us to remove that 2-meter distance. So I think the government guideline did say that it is optional for the face masks itself. But I've got a good idea what Karen might be about to say. Karen?
So maybe with...
Oh, start again, Karen.
Yeah, maybe with face masks, we also need to consider people that lip read or are hard of hearing, because if you stand 2 meters away and shout, then it's not really going to help their understanding of what you say. So I think we need to consider different users and different audiences to what we're trying to say. So that needs to be considered as well.
Thank you, Karen.
Indeed, I think that overlaps a little bit with the training. If we are going to be doing any face-to-face training, it's going to make it much more difficult if that's the norm.
Karen Underwood makes a really nice example about the US of having, and who's responsible for providing that PPE. I know some institutes, I think Rockefeller is providing masks at entry, I think, and other sites they're having to be required to bring their own PPE. So Bangwin?
Yeah, so I guess that I have to say that I wear masks all the time, just purely because of the cultural background. In our Asian background, that's wearing masks for 2 reasons: that you can protect yourself, that you don't inhale any aerosols. But at the same time, if you would be asthmatic, that you don't have symptoms, but you have the virus, you protect others by covering your face. So this is for the 2 reasons. And I still think personally, this personal opinion, that for the UK government didn't put it in compulsory, is that the stock is not there yet. So we may risk to fight the resources against the NHS. And for GSK, I can tell is that we will issue the masks for people for traveling in the public. And we had internal discussion about using face masks for doing experiments, because a lot of us were handling PBMCs or blood samples. And for that, we do say that even we're going to wear masks, but then the problem is that if you aerosol generated in the lab, the people in the lab also needs to wear masks, even they are not doing flow cytometry work. So that will be a problem because we are short of the resource. So still the fundamental principle is trying to conduct your experiments within a biosafety cabinet class 2, or you inactivate your samples. So this will be the priority. Then wearing PPEs like gloves is for sure, you will do that. And with the washable lab coat or changeable disposable lab coat will be good. The mask is still put there in a question because that will be safe as a last resort.
Yeah, thank you. It's a nice point about the washable lab coats or wipeable lab coats, because of course a lot of lab coats are cotton, which, unless you're going to wash them between labs or rooms, are quite a good way of transferring particles from one lab to another lab. But then at the same time, I guess the lab coat's going to protect us from carrying what's on our lab, what we catch at work, back home. I think by default, our science labs do protect us to a great degree in our normal working practice. And flow cytometrists are usually quite good at wearing lab coats and lab gloves. A lot of the talks yesterday for electron microscopy and light microscopy, yeah, they generally don't wear gloves or lab coats most of the time. So this is quite foreign to them. Whereas I think for flow, it's common practice. We're using a lot of wet solutions that can contaminate our clothes, and that's what we want to avoid. That's also protecting us.
Yeah,
so Joshua has come up with a point that is the non-N95 masks don't protect you, and I think that's an important thing that people need to realise, is that there's different levels of what actually is classed as PPE. So the masks with filters that the doctors are using in hospitals are true tested PPE. A scarf around your face isn't true PPE in the sense that it's been tested, but it may still help. And also another thing is not necessarily whether or not it's going to stop you infecting people or vice versa. You've got to think about the fact that it might help people to go to work. It might make them feel more confident to go back to work if they're wearing a mask. They might help their mental health, that they feel that they're not necessarily going to walk into a workplace when they've been at home on their own for weeks and suddenly get infected. So maybe wearing a face mask and gloves is also a psychologically positive thing for people.
Some nice points by Eric just there as well about the lab's general safety is pretty much where it's there already. A lot of those health and safety, and yeah, we can't be using N95 masks. That's unless our work dictates it, we really shouldn't be going near it. Bangwin, and then Chris.
Yeah, absolutely. I really think that people need to have a sense about what's the different degree of the masks we are using. So FFP2 is the equal one in Europe, or in the continent Europe and UK, about the N95, like the US standards. So those are for people who really needed to be in close contact, in very high danger when you go to visit the hospital or whatever. But FFP1 is also 80% of the filtering. And then we need to understand also surgical mask is filtering the air get in and then filter out the air getting out. But all those FFP2 or 1 or 3 that you can get from those garden centers is actually filtering the air getting into you, so getting out to you. So you need to be careful what mask you're wearing. But the bottom line is that if you just doing your lab work, I don't think N95 or FFP2 is really necessary for that purpose.
Thanks, Bangwin. Chris?
I think the most important PPE is probably strong management, and actually making sure that researchers know if they have any sort of illness, that they shouldn't come in and do the work. There's a lot of pressure behind the scenes and pressure placed on themselves, especially if in the middle of an experiment, to come in and finish it. And we know what happens. But so I think it's really strong that the management, that the bosses of everybody, let them know that then they shouldn't come in, and it's okay for them not to come in and not to finish an experiment if they're ill.
I would echo that strongly. I think actually it becomes socially inaccessible. Oh my goodness!
I'll translate. Socially unacceptable.
Thank you, Derek. If anyone seemed to even have a minor sniffle and be turning up at work, which I guess we should always be that respectful, but you know, most of us work through a sniffle. I think there'll be a big social an acceptance of that now. Alfonso.
Yeah, thank you. So the point that we have discussed as well on the mask is the need of wearing them when you enforce at least 1 hour between the users and social distancing of 2 meters. They will not be interacting, so what's the reason for wearing a mask?
I was just distracted by some comments.
Yeah.
I think I was just looking at the lab coat debate that's going on. If we were to provide lab coats for every user, there would have to be one for every user that comes into the Flow Lab, which is quite a lot of users. And if they're disposable, that's a lot of disposal to also get rid of.
Yeah, and the mask goes in the same direction as well.
Yeah.
Mask, and there's really no need for that.
So I think the mask, I think, is always going to be quite a polarizing view. There's quite a few saying no, we should definitely have masks. Some saying not, coming up on the chat at the moment. It is polarized. It's interesting because when we walk into a supermarket, we have to wear a mask, but when we're anywhere else or asked in the UK optional, I think it is going to be what we are guided to nationally and locally, the best way to go with that.
You know, in the supermarket you're crossing and corridors are not quite broad. But here you're saying that you have to guarantee 2 meters distance between users, and you're putting as well time distance. So this is slightly different than the scenario in a supermarket, and you are reducing these consumables that at the end of the day they will end up on the bins as well.
Yeah. I think the chance of someone coughing and spluttering onto the keyboard and the cytometer that we're going to be using is quite high because they can be there for prolonged periods of time as well. Marta?
But then you will be cleaning afterwards, Peter.
Yes. Yeah, I think you have to. Marta?
Hi, everyone. Well, concerning the masks, I think it depends really on the general policy, because masks will be more effective if everyone is using them. If the general policy of the institution is not to enforce the general use of masks, well, of course, their use and importance will be relative, right? And for us, the policy is that inside the institute, everybody must use a mask. And I think, and of course, keeping the distance, that is really critical.
I think we'll be coming on to that distancing at the next topic. Actually, we're almost getting there to that next topic point.
Yeah, there's been quite a few points in the chat about testing, whether institutes are offering testing, temperature testing, or even PCR tests.
What's happening at Crick, Derek? There's loads of rumours flying about.
As far as I'm aware, there's other people from the Crick on this call. I think the plan is once we reopen to offer weekly tests for staff.
That's voluntary or compulsory? Do you know?
I don't know whether that word is in the sentence. Hang on, Andy might be able to answer. He's just raised his hand. Andy?
Whether or not it'll be
Andy.
voluntary or can you hear me?
Yep, start again,
yep.
Andy.
It's going to be, it's still in decision about whether or not it's going to be voluntary or mandated. It's probably that we would be wise to get tested. We're also working on a serology test as well. This will tell us whether or not the antibodies are still are protective or not if you've had COVID. But the secret here really is about testing and a good testing regime. If you have that in work, then the surety goes up, your own personal surety goes up. So that's what they're doing in the Crick anyway.
Yeah, that last bit is a really crucial bit actually, not the actual testing. I think for someone like University of York, we have so many other types of departments, not just biology, that we could never test that number of people unless it was a national facility that it was going to. It's not just, obviously you've got all the other sciences, all the arts, humanities, and everything else. This is being recorded, so I better be careful what I say on that front. But it does make a difference of, even if we can add just a few extra. We're seeing here, some people say masks, some people say not masks, some people say gloves, some people say not gloves. What would be really good is actually those who are very worried about it may not feel confident to walk into our shared labs because they don't know what the last user has done. And I'm quite concerned to make sure that all users feel safe and secure to use the lab, so we don't minimize the number of people. So it may be more than the rest of the department or institute is requiring, but at the same time, having those extra elements, even if it's just available for them to use, it can make them feel confident coming to use our instruments because it's not in their own lab space itself.
Could we bring Andy back in?
Yeah, I mean, I think Rachel said it there, when people feel confident within the thing. The only way you're going to get really true confidence is if you have a testing regime in there. I mean, you can go through all the precautions and everything and look for the risks. As I said earlier, the biggest risk will be people coming to work because there you have no control. At least within your own lab, you can put in specific wearing gloves and wearing lab coats and so on. You can do that. But yeah, testing is the key here and getting that up. And the CRIC is in a fortunate position in that it repurposed itself for testing as part of the national push anyway in the UK. So.
And they're expensive. Those tests are very, very expensive for the RNA test. Telma, thank you, Andy. Telma?
Sorry, I was listening to Andy. No, I was just thinking about these masks thing. So we have the mask mandatory in our institute, but also I think as a facility, like we were discussing before, we need to think that we don't have the same people in the lab. It's rotating, I don't know, like 20, 30 people per day in the room where the air, it's not so well ventilated. So I think we also need to think about it. And I think the masks, okay, we are using the serological ones, so they are not super protective, but they help and they prevent. And I think that's the best we can do now. It's to prevent.
Yeah, thank you, Telma. Joshua?
Something, how they described it as Schrodinger's virus. You have to, everyone is both infected and non-infected at the same time. And until at least in your locale, you have a testing regime like South Korea where everyone can get a test on demand, it's worth treating everyone as if they could be asymptomatically infected. I think that's how we should just treat PPE. So it's about you stopping spreading whatever might be on your person. I think that's the most effective way, because obviously the hospitals require the PPE the most, and so if everyone stops projecting any droplets, the need for face shields is significantly decreased.
Yeah, thank you very much for that, Joshua. I don't think there's a comment as well. For working with COVID patient samples themselves, which I think is a completely different type of question than actually working with colleagues that might have the virus. Actually working with the virus requires a whole set of different start procedures, at least. Once they're inactivated, then there's not such a big problem. There's a conference coming up at CRIC, I believe, Derek, by Lucy Collinson. I know it's electron.
Next week, I think. Yeah.
But I'll find the link for that. And if you are working with COVID samples, there's a big discussion, including some of the Cat 3, Cat 4 labs nationally that are involved in that type of work, sharing their top tips and tricks and different things to consider. I'll find a link in a moment on that. Are there any other thoughts on this? Double gloves? I find it hard enough to get one pair of gloves on. Yes, I'll find that.
Okay, sorry, Karen.
Karen?
So just seeing there's some asked questions on the chat about for doing COVID-19 testing and what was needed. We're doing heat inactivation of the patients or the suspected patient samples at Cat 3 and then moving it out into a Cat 2 to do the RNA extraction and then the RT-PCR in separate rooms. So it's needed initially, but then not onwards. So just to clear at that question. I think a couple of other people have suggested the same on the chat.
Yeah, so you've been doing a lot of the testing at the hospital, Karen. Thank you very much for that.
Yes,
Yeah.
Right.
keeps me out of trouble.
Yeah, yeah. I would like to comment on the lab coats actually, and also on the gloves. The lab coats. We had a discussion today in the technical department, and one of the options is to autoclave them as well, because in Germany there is hospitals, or there is a guidelines about the FFP masks. They can be reused if you keep them overnight over 80 degrees. And now there's a discussion about lab coats, maybe can be put it in an autoclave bags and then autoclaved, and then maybe daily or once a week or every 2 days. And that can be an option or solution for the lab coats. I mean, depends on the number of the users. And my suggestion is regarding the lab coats of, I wouldn't recommend to ask the users to bring their lab coats, because you never know how they are kept, their lab coats, and how safe they are, and if they are clean, or how they take care about their lab coats. So maybe asking them to bring their own masks, but the lab coats should be provided maybe by the core facilities. That's an idea.
I think, Rafe, with people well, the government is recommending that people need to cough, they cough into their elbows. So if you have people coughing into their lab coat elbows and it's a communal lab coat, then well, that's how we're going to potentially spread. So you're right, maybe masks are more important than a clean lab coat each time.
Rafa, thank you very much for putting for that web link on for imaging SARS-CoV-2. Thank you very much for finding that for us and bringing it up.
Jane's had a hand up for a while.
Oh, hello. Yes, just a quick point about the people, the health of users. One thing I've sort of heard mentioned when it's not possible to test everybody is that people sign a statement to say that they're healthy, to make people really think about their health rather than just sort of assuming that people have thought about it. So that could be an intermediate way just to help people to really think about their health before they come in.
That's quite a nice suggestion.
They're suggesting in our place that people have their temperature taken as they enter the site, or other places doing the same.
Andy?
No, he's disappeared.
Yeah, but I say he's not usually slow at speaking, is he?
Got it. Sorry. Just to follow up what Karen said about doing the genome testing. On serology testing, at least, it's CAT2, because it's not so much a COVID, it's more to do with what other bloodborne viruses are in the sera. So it's just another level of protection that you have to go through.
Okay, should we look at the polls?
There we go. We'll have a couple of polls at the end of the session.
Okay. So if you could go for the polls, please.
Yeah, they should be able to see them. So Adam's opened that up, I think.
Thank you, Adam. Yeah, thank you once more to the Royal Microscopical Society, Adam and Victoria, who are working behind the scenes, keeping this moving on, getting entrants as they come in, keeping these polls going and collating the data as well. Okay, that poll seems to be coming close to an end now. Just a few more coming in. And should we close it there, Derek?
Yes, why not?
And share results. Can anyone copy and paste that? Adam, do you get a hard copy of that result you can put into the chat so we have a record of that for everyone as well? So quite clearly, should users wear gloves? I think that's overwhelmingly yes. I'd like to compare that to the 2 meetings in the days before. We'll get a summary on that and we'll share that results with everyone as well. so you can see how that compares. From my side, I'm in agreement, but it's an interesting one. Sorting, we never talked about sorting actually. I don't let my sort operators use gloves on the keyboard because the chances are they've touched something that's Cat 2, Cat 3, put it into the sorter, and then they should remove their gloves before they use the keyboard. Otherwise, the next time they use a keyboard, they might not have gloves and cross-contaminate themselves. We didn't bring that point up, actually. That's one area that I would not want them to wear gloves around the keyboard. And I'm quite 70% happy with wearing masks, 16% unsure. That's also good. So actually, just before we go to the next session, sorry, Rachel, what's everyone's thoughts about when you're using gloves to stop contamination from your sample and then go into a keyboard? I mean, for us, that is an absolute no-no. In our health and safety, we're not allowed to do that because we can contaminate the keyboard, and someone could just walk in then without knowing and cross-contaminate themselves. What are the thoughts on that? Karen Hogg.
So I do get a little bit nervous when I see users with gloves on using the keyboard, and you almost, want to question, have they just handled their samples and just put, contamination of various different antibodies, DNA binding dyes, various different samples that, we don't have all of our samples fixed. So many of our samples are unfixed. And so I think it'd need a little bit of education to the users, and well, maybe also to my mindset, so I don't question them every time I go into the room, to using gloves with a keyboard. So yeah, I'd be a little bit cautious to that and need a change of mindset. So that's my comment to that. And yeah, we don't use the gloves with the keyboard for the sorter at all.
This is beautifully divisive in the comments.
Yeah. Pete, you've obviously caused a flurry of hands. Eric.
Hi, good day. Yeah, so with our sorter room is specially auto door swipe access limited room. So only operators have access to the rooms, and any users who are bringing samples in to us or users who want to talk to us, they will have to wait outside, or they'll have to press a doorbell and then we'll let them in, kind of thing. So anything that's, and users, anyone who's coming in, we let them know that all surfaces are potentially contaminated. So if they want to touch anything, they have to wear gloves.
Yeah, I noticed Chris Hall, Chris's comment about gloves just put it beyond doubt. But I'm very, so we do work with some high risk category organisms, and really once you've touched the tubes that they're on, you have to remove those gloves at that point. We could always put another set of gloves on and carry on working, but at that point the area outside should be safe. And if it isn't, then we're not doing our job properly. So it makes sure you are stayed on the ball at that point. I think it's quite an interesting point. I don't think there is a clear-cut answer. I wish I had another poll now on this for that scenario, because I'm going to be I would rather wear gloves now when I go to a keyboard and a shared resource, but it's for a completely different reason.
Yeah, exactly. The focus has shifted.
Yeah, very much so. And
anyway,
before it was quite frowned upon that you should.
Yeah, we have another hand raised there, Pete. Thomas.
Thank you. Sorry, Derek, I missed that.
Well, I actually completely disagree with changing the gloves or taking the gloves off after handling the samples, because when we watch our users, there is this high risk that they actually do it accidentally wrong. So what we have instated now is that they wear gloves at all times, and we have keyboards that can be disinfected and washed, and which are fluid proof. So there is no difference between the dials or handles of the flow cytometer that you have to touch and which you can disinfect, and the keyboard that you can disinfect afterward. You just have to be disinfecting both.
Yeah, I fully understand that. I think some of the organisms we're working with, which are Cat 3 organisms, we can't take that risk of spreading it anywhere. We're trained, the cleaners aren't. And the cleaners can come in. It's not just experts in the lab at all times. So that's what we chose locally on that one. But it is quite a lot of mixed opinions.
It is. It's divisive. Bangwen, you have another comment?
Yeah, I think that I would also intend to have the keyboard that you can disinfect, but while the gloves all the time. That's what at least what we recommended in GSK, that we will always wear gloves, even we are just typing with a lab-based computer. But then we have those plastic cover on those, so you can disinfect the surface from time to time. So I guess that can help, because you don't want inconsistently taking off your gloves and then putting on new ones, and then you can just keep consistent working with the cytometer with gloves, and then touching your keyboard with the gloves as well.
Okay.
Yep, thanks, Bangwen. I think we should probably move along. I think there will be some overlap, and we'll be coming back to some of these PPE things in the later sections. But we wanted to move on to topic 3, which I think Rachel's going to handle to start with.
Okay, so we're going to talk about physical distancing. So the government, or the UK government, I should say, because I don't know if it's different in different countries around the world, have recommended that we keep 2 meters apart from other people unless they were within our household. So what rules will you have in your core facility? Obviously, we want to keep the physical distancing of 2 meters. But how's that going to impact with the machines in your lab? Does your institute ask you to have 2 meters apart between machines, and therefore you're going to have to move them? Or do you have restrictions on the number of people who can be in your core at any one time? So I'll open this up.
And you were very quick off the mark there.
Anne?
Hi, Anne.
Yeah, I've got video. I haven't got video. I was just trying to put it on, and it doesn't seem to. Anyway, it doesn't matter. Start my video. Hi, everyone. Well, we've been back at work in Lausanne at the university for 2 weeks, and we had a week or more to set up. And what we had to do was rezone all of our labs. So we have one that's 40 square meters with 6 machines, which is quite tight in space. So we rezoned this to 3 zones, 2 machines per zone. And on our booking system, we actually set it up, this is in Iris, we set it up so that if you book one machine in the zone, the other machine, it's not possible to book it any longer for the same time. And it's an add-on in the progress. We have one other room that has 3 machines, and it's only 15 or 16 square meters, and that's 1 machine out of the 3 at any one time. And this gives physical distance of 2 meters or more between anyone sitting at the machines in the zones. And so far, it's working okay, but it has obvious consequences for revenue. So we're highly encouraging the lab, the people who are working, the researchers that want to use this is the analyzers, people who want to use the analyzers to work shift work, longer hours. For the sorter room, we have 2 sorters in a room that's small, so one operator for 2 machines at the same time, or only 1 machine. And in that respect, so far it's been quite quiet. And this is seeming to work so far so good, but it's early days on this. We haven't had any complaints either, which has been good.
How are you prioritizing users?
We haven't had to. It's a kind of first come, first booking service, but the thing is, all of them, we have mostly people working with mice, and they were all had to kill most of their mice 2 months ago. So they're still setting everything back up. That's why we think it'll be a gradual return. And my staff are working the same, they're working 8 till 2 and 2 till 8 for sorting, with a bit of flexibility at either end. They're all keen just to go back to work. So for the next few weeks, I think this is how we'll proceed.
Do you have any issues, Anne, with people, if you've got machines in the same room, where people have to break the 2 meters to walk behind somebody to get to another machine?
This is a bit of an issue, but they're encouraged to be quick. One of the rooms you have to walk past almost through the first zone to get to the other zone.
Which should be okay as long as they're not coughing or sneezing.
Yeah.
And
they're wearing masks and gloves and disposable lab coats before they even enter the room. And as I said, we haven't even had to put time between the bookings on the analyzers because it hasn't been that busy. And we know that some of the researchers are working late in the day, so they're running samples at night. Okay.
I think Thelma wants to jump in. Let me just. Thelma.
Hi, I just wanted to comment because we have similar to Anne. So we also have this online system that we can block the machines, and when one is occupied, the other cannot be used. And it was working so far, but now that the labs are also more, so we can have like 4 to 5 in the same lab. It starts to be very complicated. So now we could keep the 2 meters distance so far, but now we have like the professors complaining that they cannot use the machine, they need more time, and we are not sure how to do now. If we should put like a physical barrier between the machines that are too close, or how to do it now. But I don't know how you guys are thinking of doing it.
Has anybody put physical barriers up between machines, or has anybody chosen to, if they have the space, to expand their core out into other rooms or other buildings?
We did that. We also did that, Rachel. So some machines we also have to change, and some of them, even for tests, we could manage to put a little bit more far, but there's a limit. And now, yeah, I'm just curious also how others are going to do it.
Okay, we'll bring Andy Philby back in.
Yeah, hi. So I was going to say one of the things we're trying to do, and it may be completely unsuccessful, is we're trying to push back onto the research groups and say to them, you know, we're trying to operate in a way which is supposedly intelligent, and we're splitting our group into teams so we have skills that are not overlapping. We keep people with certain overlapping skills physically apart from one another, running in, you know, different teams. But really, the research is going to have to work in a similar way. So, you know, it may be that we can minimize footfall into our facility and risk into our facility partially by, you know, saying to groups, you know, if you're a big group, put one person in who's going to come in and do all the flow, or, you know, if you're a small group, group up with other people. But these PIs also have to think of ways to, you know, for them to be operating under these conditions. It can't just be us doing it. Otherwise it becomes, you know, it's not going to work. Like Andy Riddell was saying, you know, we can't control travel into the institutes. We can't control a lot of what's happening in the policies and the movements around in the corridors outside our facilities. But one thing we're trying to do is we're trying to push back on the PIs and the groups and get them to think about this as well.
Yeah, excellent point, Andy. I like, you know, that's the first time I think I've heard the idea of having super users that can be nominated for a lab to minimize that crossing over.
Yeah, I mean, the other thing as well, we'll get onto training, is we can't be training undergrads and students, but their projects may be quite pressing. You know, as a university, we have people wanting to complete, we have a massive commitment to education. And one of the ways we may be able to get around that is to use a model, actually, when I was working for Derek, is that, you know, we may be accepting people's samples for things like cell cycle analysis and the simple things that we can do easily, and maybe just offering that as a service. So having super users for the complex immunology, complex panels, but trying to push through the other stuff as our service.
Yeah.
We'll
I think some of that may come up again in the last session. Yeah. So
Okay.
Bangwen.
Oh, sorry.
I was just thinking that maybe I already hear a lot of nice suggestions. But another thing I heard, I think, from the light microscopic session is that would it be actually practical to create arrows on the road maps that's to guide people to enter from one door and then exit from another door, so you don't have the cross contact with the people, and then the workflow would be in a more, you know, in directed way, just like when we do shopping in the supermarket. So in that way that you avoid that unnecessary contact with people, face-to-face interact.
Okay, Pete, you've got your hand up.
Yeah, absolutely. I appreciate that some labs are at the end of a corridor, and so they can't have a one-way system. And obviously some labs, quite a lot of labs, only have one door in and out. But just the corridors in and out at York, we're operating a one-way system in the corridors, so we can get out either end of the corridors, if not necessarily the rooms. And if you can do that in the labs as well, all the better. It is a good way to minimize contact. Sorry, okay. Rachel.
We'll bring Thomas back in again.
Hi again. I just wanted to put into the discussion that it's not only the simple distance that is important, it is also the quality of the room. So one important factor is how often the air is turned over in that room and whether or not you have windows that you can open to get some airflow going through to basically push out potential aerosol. So this is quite important. So what we actually did is we went through all our rooms and we have defined a maximum occupancy on all the rooms and have adjusted our reservation system accordingly so that people can use these rooms only with the maximum numbers that we have allowed there.
Yes, there's a good point in the chat there about the fact that many places are only operating at like 25–30% operation at the moment, and what works now may not be something that works when we get more and more people coming in. That's something we're going to have to bear in mind.
I was talking to someone from Austria this morning, and they have limited to about 30% at the moment and working in teams, but I think either this Monday or following Monday they're back to normal. So it's quite a short lead time to being almost completely unlocked, and I can imagine users not feeling confident yet. I think we've got a bit longer than that, certainly in the UK, but some countries, the unlocking can be very, very rapid indeed. It's keeping user confidence. That's important.
A question that's come up on the chat from Alex of whether or not people are allowed to work back to back. So I know that some places have machines, analyzers that are back to back, and therefore you're not, are you sharing air?
Back to back may be better than side to side.
Yeah, so if they're working back to back, so they're facing opposite ways, I know that that is strongly encouraged in our unlock documents. We're looking at back to back working, but we're still looking at distance as well. What about how long a room should be left before the next user goes into the lab? What's people's feelings on that?
I guess that also means it depends on if you've got a call with core staff and they have to walk through to get to the sorter lab, you're always going to have potentially a member of the team and a user. But then if you are relying on money coming into your core, time is money. So to have an hour between is an hour when somebody can't be doing work, you're not necessarily bringing in an income. Is that something else that perhaps needs to be taken into consideration if you work in a core where your income, I know this is difficult times, but if your income is dependent on, or your staff numbers are dependent on your income, then do we need to try and maximize our income and allow people in and maximize the time on perhaps just one machine as opposed to several?
I think there are so many different people on this call that have different models for that. It might be difficult to come up with something that's one size fits all, but you know, certainly as you say, Rachel, these are odd times, aren't they? And we're probably not going to be operating to the same level and constraints that we were beforehand. It'll be interesting to hear from other people here.
Yeah, looking at the University of Berkeley and they're from Brith, that's quite good. It's one person per instrument, so don't bring a friend anymore.
Yeah. Katie, sorry, you have a comment? It's all yours. Hi, Katie, we can't hear you.
I'll start again, sorry.
There we go.
So we have two instruments that are back to back in quite a small room. They're probably about 1 meter apart, or the users would be 1 meter apart, and we've moved the max quant to the far end of the room, so there's at least 2 meters social distancing. And because the max quant is fairly automated, we'll be asking users to leave the room, you know, during startup, and then when they're actually acquiring samples, they'll have to use like the plate reader or the chill rack 5. So it limits the amount of time that 2 people are in the room together. And the air changes are about 7 per hour. So I don't know if that would be enough to protect them. Did you hear me?
Yes.
Sorry.
None of us have got an answer.
Yeah. I think that's a difficulty. I mean, they will be 2 meters apart, and there'll be a brief time when one has to pass behind the back of the other. But apart from that, the only other thing we could do is only have one occupancy in that lab. But then, you know, if the other person is only going to be in the room for less than 15 minutes or 10 minutes, then the shared space is quite a short amount of time.
Yeah, and it depends on the airflow of the room itself.
Mm-hmm.
Again, my argument for the room is we don't know how long someone's just left the toilet.
Yep.
And yet we can walk into the toilet straight after someone in theory. So we're looking after our labs to a much greater degree of caution to give our users confidence. And yet in our everyday life, walking into a small shop that will be open, walking into a supermarket, we're much greater risk than what we're putting in place at the moment. And I think we're going to have to trust users. I say we're looking at a 10-minute rule, I think, at the moment at York, just for good practice. But how do we monitor users? How do we know when they left, when the next one walks in?
Generally, we would do that through the logbook. So each instrument has a logbook. You sign in and sign out.
Is that on the outside of the room?
No. Yeah, that's true.
It will be. Yeah, it will be. No.
Not in the virus labs.
Okay, we're thinking it's exactly the same thoughts on ours. How do we check that? And we thought a logbook outside, and they should have lab books, so they should have their own pen to write on it as well.
Mm-hmm. Yeah, or potentially you could have a whiteboard outside with someone. You know, you write the time, you log out, log in.
People, when they have got people coming to do sorting, are we, I assume people are letting people put the samples 2 meters away and then letting people stand 2 meters back to be able to watch the gates being set up? Or have people been able to put TeamViewer on their computers? I know in our place that IT don't really like us doing TeamViewer. So we haven't got that on any of our sorters at the moment. I'm wondering if that's something that we need to insist on and then have a monitor, even perhaps in another room, so people can help guide with setting up gates.
I mean, what we were going to do for our virus sorts was just have the user bring the samples in. Either they can stay with us whilst we set up the gates and the compensation and then just say they're happy with the gates, or they could just stand 2 meters away, try not to breathe heavily over us whilst we check their samples. So that's what we do.
Which very much comes to the last topic, which is that staff user interactions, which we'll get to as the last topic today, because I think that's quite a hot one.
Yeah, and I think we can cover those sort of desktop apps and things in there.
Has anybody put up any barriers between their instruments in order to have a physical barrier so that you could potentially be within 2 meters? Or is that something that your institute doesn't like? I know some offices are looking at putting up barriers between. Anybody doing that at the moment?
Can I?
No.
There was a sub-conversation going on just while you're digesting what's in the chat. Andy Filmby asked, or thinks that we should get discounts or some rebate for our service contracts. Do you know, I thought long and hard about this, because actually I was going to go start asking, just as Andy has done, for service contracts. And then beautifully, you've got to think that actually they're still employed, unless they're being furloughed, which is a different argument to that point. But if they're still being paid, they're still there, it's still liable. But also, when we go back, the number of instruments that will not start up when we go back will be far greater than normal. They are going to be really rushed and stressed, and we need them to keep their service team and not start making them unemployed because they've had 2 months of not being able to work. So actually, I kind of felt myself being quite empathetic towards the service engineers I'm quite fond of, and I'm still paying their salary. I am asking, though, not to have service contract price increases, because inflation has dropped and we shouldn't be seeing price increases on service contracts. And I'd much rather see that benefit. I can see Andy's chasing my tail here. Go on, Andy, go.
Well, no, I do agree, and I think it's important. It's not about the service engineers; they're wonderful, wonderful people, and we all love them dearly. But I think when it comes to, you're right, when we come to negotiate our contracts every year, as many of us do, and probably enjoy that moment immensely, we do get a lot of these conversations about how many call outs have you had, how many times has this failed, and these metrics are all built into the price that comes up with at the end. So I do take some just moment here to say I'm not saying we ask for 100% or 50% back or anything, but I do think there needs to be a little bit of thought paid by the companies about yes, they do have engineers, yes, they are coming out for critical work, but the call out rate is dropped, and that will have an impact on the costs that they will be having to deal with. So fewer parts being replaced, fewer visits, fewer trips. So I hope that is definitely brought into mind. It may be we all go back to the labs and our machines are all, full of PBS crystals and things are all wrong. But, if we did a good job of shutting them down, I'd probably say not. But let's see. But I do think the companies need to be a little bit mindful of this when we come back, because as Rachel pointed out a few minutes ago, if we're on cost recovery and we're having to operate in different ways, what do? Do we put the costs up then? Do we take the model that, we've discussed in the past, Pete, it costs what it costs, you've got to recover what you've got to recover. And we're not going to be able to do that because money's down for research altogether. So we have to be a little mindful. I think the companies are going to have to take a bit of that pain too in the servicing.
I think Anne wants to jump in here.
Yeah, just quickly for Andy's comment about the machines. When we had to shut the lab with literally 1 day's notice, we, over the first week, my guys went in one at a time and filled up almost all of our machines with water, took all the sheath fluid tanks and everything apart, all the sips off the analyzers, did sterile shutdowns on the ARIAs, and there was a suggestion from someone to fill an Astrios with ethanol. So we left them sitting for 2 months like this. And when we came back and restarted everything 3 weeks ago, everything worked fine. Just to let you know, it is possible that your machines might all be fine. But we were worried about PBS crystallization for things that were sitting there not being used for a long period of time.
Yeah, and I agree. I think the days of gas lasers are gone, where the gas lasers need to be, or used to have to be frequently turned on. Yeah. And pumped through. That mostly solid state now, which negates that side of it as well.
Yeah, yeah.
So our lab hasn't closed down fully. We're still working on critical experiments only, and we had a few outstanding engineer visits, and we've actually allowed the engineers in to do them because we knew there weren't going to be any other people in the lab. So we timed it in with when people weren't on the other machines. And one of the reasons we've done that is because we know that when everybody gets back up and running, engineers may be in hot demand. So we thought we may as well, whilst we've got the parts sitting in the lab, we may as well then have a working machine that's then shut down.
Rachel, that raised a really good point as well. I think I wanted my machines always to be ready to be used. One or two have been used over this period. So I didn't want to draw them off contract either. So I wanted to make sure if they were suddenly needed for any essential work that they were still there and still under cover. So I never went back to the company and said, please pause your contract on resume, a bit like a gym contract. If you're at the gym, you can say, I want to pause my contract, I'm going on holiday, come back, start me back in a month's time. So I didn't tell the companies. I think that they will, there's going to be companies listening to this, aren't there? They might try and squeak out of it by saying, well, if you'd have told us you didn't need it, we could have paused it. So that's an interesting question as well. In which case, I'm going to pause it for 1 week over every Christmas, by the way.
We have another comment for you too.
Hi, I can start my video. Yeah, hi.
Okay.
Can you hear me?
We can hear you.
Start my video somehow. I think, yeah, it's been stopped. So, okay.
Yeah, got you. We can see you.
There you are.
All right. Hi. Yeah, so this is, I'm from Vancouver, British Columbia Children's Hospital. So similar to Rachel, we also didn't have any really fully closed down of the facility, and our machines were still in operation for a lot of critical experiments that happened. And also, we also had the engineers' visits as well to our facility. Just as she mentioned, we were also thinking about how they would really get caught up with multiple requests right after the facility really opens up for everybody. I think one of the points Rachel mentioned earlier about the income being one of the concerns, which is, I think globally, like everything is being affected. So we did also want to have the service contract sort of being pushed up like a little bit longer. We discussed that with our vendors as well. And similar to us, they're also kind of facing the same sort of, economic constraints as well. I think it's a common scenario everywhere, and for now we are just leaning more towards the financial support from either institute, university, government to kind of talk about our losses and kind of get some support there. But at the same time, we are like safety is kind of the priority for everybody. So I think even when we are discussing about, do we want to put the gaps in between the users so that there is enough time and people don't run over each other? Or do we want to, kind of bringing in the engineers in, or kind of all of those decisions are being made with regards to not necessarily revenue loss or, what's going to happen, but it's more to consider like safety first sort of thing. So I think that's something which I wanted to mention, that there is a loss that everybody is understanding, but it's bound to happen. And we just want to be like, we're considering mostly the safety of the users and the staff while making those decisions.
Has anybody else got any thoughts on the time that we can have between users? Maybe we can put the poll up now and then we can see what people's thoughts are.
You've got enough time to read those.
Oh, damn, I forgot I can't vote. I'm trying to vote, like, I can't vote.
You just need to cough, Pete, where the answer you want, people will vote for you.
Yeah, so careful. So the question
don't forget to
that are not accessible at any time. So how many instruments will be offline at any one time? That question too.
Also, don't forget there are 3 there questions, and you may have to scroll down to find the third one.
I think this is actually different to the LM results.
First question certainly is.
I think the third one is coming out different. We'll have to share that and just talk about it briefly. But first one's massively different. Okay, just get those last votes cast, please. Shall I end it there?
There we go. Yeah, that's probably as much as we're going to get.
So it seems that in flow cores, we tend to have our machines all in one, or in most of our machines in one room, which I guess is different from LMP, as you see.
Yeah, we're fortunate we got quite a lot, but yeah, there was quite a few individual rooms for instruments. For LM, more so than this. So a lot more shared space for flow. A lot bigger problem, therefore.
And most people are going down to probably about 50% of their instruments. So,
I think that's quite a high percentage still in operation.
Yeah, but I think this is a really important figure to feed back to our funders, national funders and our institutes, because it shows just how much research can't be done with the social distancing that's in place. And this is the impact on the research, how much research output can be done by our research labs.
Exactly.
So if they come back, yeah, quite a big impact.
Considering how important the core facilities are in most research, there's going to be a bottleneck there.
Hopefully not everyone is going to want their own system after this.
That's a possible consideration, of course. Maybe we should add that in somewhere.
How long should the room be empty between users? The majority is saying 15 minutes. There's been something on the chat about air changes and how in 15 minutes you could actually have quite a few. As Katie said, that it's 7 air changes per hour, so 15 minutes should be long enough. I've never considered air changes, but that's something I will ask our engineering department.
Yeah, there's a recommendation of at least 2 air changes on some countries' recommendations. So yeah, that would add up to a 15, roughly, in that ballpark. But again, toilets. I wish the air changed much faster in the toilets, if I'm honest.
And also, it depends how many machines you've got in a room, surely. If you've got half a dozen machines, are you just going to restrict that to one person in that room? And with a 15-minute gap for the next one?
Yeah. And that then brings you to Will says there's loss of revenue. And I think, all universities accept there's going to be loss of revenue across many areas. University conferencing facilities, things like that, and catering are huge losses, much greater than our own. We're actually relatively small at that point. We just have to make sure our line managers and everyone understand and manage their budget expectations.
Okay, I think this is a good point to pause for about a 10-minute break. So we'll come.
Yeah, if we come back at 5:02, should we say that?
Yeah.
Yep, if we come back at 3:55, so everybody's got a chance to, have the traditional cup of tea and bathroom break. And we'll come back and cover the last 2 topics in 12 minutes time. Obviously, if you think of any questions, carry on using that chat while we're away.
Thank you, Adam. If you could mute, and that's all. Hi, Rachel.
I was just closing my window because rush hour is starting. I'm not used to traffic.
Yeah, not just for the air change, so you're starting to think about the air changing in the room.
Yeah, that's true.
Right, this one might be take a little less time than the last point. I think it will start to move into staff using interactions naturally anyway. So I'll try and keep it very focused on our staff issues and keeping our teams present.
Are we talking to everybody now?
Yes.
Yeah, anybody who's here, yes.
Okay.
It's not just us. Yeah. Okay, that's 5-2.
It is indeed.
Shall we reconvene, everybody? Hope you've all got a cup of tea and are suitably rested after that short gap.
I just went and got some birthday cake.
Don't forget to share it out afterwards, Pete. Anyway, the last two topics are really less to do with the actual minutiae, I suppose, of running the lab, with have amongst ourselves and with our users. So we're going to start with managing staff. So Pete, I think, is going to lead on this one.
Yeah. So this is a big one for me. We think a lot about how to keep it clean and everything else, and how we look after PPE, the physical distancing. Actually, for many of us, maybe not all, but it's still worth persisting. So we'll come on to user interactions of how we're actually going to do one-to-one teaching or troubleshooting with users after this topic. Do you know what? We've got teams and we're going to have to group them. As Andy said earlier, he's looking to make sure he's putting his teams so he's got all sort of aspects covered in one team and the same aspects covered in the other, so there's not so much duplication. But there's other things we're going to need to consider as well, such as those who have commitments with children that may not be able to go back yet to school in some cases. People who may themselves be at a high risk group or going back into their household with a member of their family that's at high risk. So how are we going to break those sort of teams down? How are we going to manage that? I think that's something that we may be put to us as individual lab heads, lab managers, to actually organize that on their behalves. The other side is once you've got that division between the two, actually there can be virtual walls that start to separate those teams, and it can be quite divisive for team morale. So I think we also need to think about now how we keep those two teams, a dialogue between them going at all times, so that these walls aren't put up. So when they come back together, we still have hopefully happy ships. I'm fairly sure that my team is, I hope, happy. A couple of them are on this, they're probably laughing, going, what do you know, Pete? But I want to keep it that way. I want to make sure they remain as a really good united team and not see that broken. So Andy, go on. I'll stop. Andy, kick off.
Yeah,
you know what I mean.
Yeah, I just wanted to sort of give a bit more background to what I said. So a few years ago, we've always had something called a business continuity plan that we had to fill in every year and update. And I used to hate doing it, but we did have a section on, pandemics. And I also concluded a section on the zombie apocalypse to ensure that people had read it. It's pretty good, actually, good planning for the zombie apocalypse. But what I did as part of that is I made this thing called the skills matrix, and I looked at all the key functions and technologies and things that we needed to support and had that kind of going down this axis. And then across that, I had my members of staff, as a primary, a secondary, or a tertiary kind of lead in that. And what it was good for as well was over the last few years, it's allowed my team to sort of multi-skill and have overlap in function. So I've been able to use that and that approach for kind of breaking my team up, fortunately, because I have enough members. Some people don't, right? To break them up into, as I said, two teams. So the idea being is that we have a lead for each kind of technology, and that will also affect what we can support, what technologies, will we be able to run the Hyperion, we'll be able to sort that kind of thing. So that's one approach I've kind of taken to mitigate that risk.
Which is good, and I'm with you, Andy. Andy, have you given much thought about, so you split the team into their skill sets and divided it with skill sets. You thought much about, I don't know if they've got child commitments or others that are at high risk. There's a potential here that one or two members of our team may have to, of teams that are out there, may have to be much safer to work from home almost permanently, until they're unlocked, until it's much safer, which means they're going to be working from home while another cohort are in the office doing their daily job, taking the heavy load. And the people who are in the office, the people who are in the lab can be feeling a bit hard done by because they're having to do all the hard work, put themselves at risk. Others at home could be feeling really guilty that they're not able to be in the lab doing that work. So they both can feel very bad about this in different ways. So I don't know if anyone's had thoughts about how they would manage that and how they'll distribute the work to make those at home still feel like a useful part of the team.
So just to kind of come back on that, Pete, one of the things we've been doing there is we'll split the group. So there will be 1 week, week 1 rota, it will be team A 3 days and then team B 2 days. And then it will flip round for the week 2 setting, and that will just rotate week 1, week 2. But what we've for a long time, where we've been working at home, we have a Teams group set up. So we have one of my team, Andy Fuller, set up a wellness chat thread on that. So that's just kind of just chatting the nonsense. It's not the work stuff, it's the fun stuff. So baking and, my piano playing, all that kind of stuff's gone on there. Yeah, I guess just having that kind of regular contact with each other, also with the team viewer and stuff, we have people at home who can dial in and actually fix problems too. And yeah, we've got the comms going. So that's a really good point, keeping that morale up. And that also, by having that split pattern working, it does allow people to also take care of, the childcare. Good point. If the schools don't reopen, then that's going to be a problem for a lot of people. It's a problem for me personally as well. Yeah.
So I'll come to you in just 1 minute, Andy. Andy Riddell, just 1 second. Thinking, do you know what? I've completely lost the plot. It's that birthday cake. It's gone to my head. I've got a sugar rush. Andy, I'll come back to mine in a minute. Go for it, Andy. Andy Riddell.
Yeah, I forgot about what I was going to say too. There, I'm getting the cake as well. It's just there was just an extra comment to make there. We have furlough staff as well. So in the UK, there's a scheme for employed people to go on sort of holiday leave, and that'll be paid by the government. I know other countries have done the same sort of thing. But they are also, so the government rules in the UK is 3.5 weeks. And they're not allowed to participate or do any work for the institution. They can, however, take part in lab meetings as long as they're not given any tasks to do. And they can do personal improvements through educational things that you can supply. But there's an emotional point in there as well where they may well feel that they're being isolated. And as you pointed out, Peter, earlier, that this thing's a very emotive thing in the round. And
I've just lost you, Andy.
Sorry, can you hear me now?
Yeah, no, you're still there.
Yeah, the whole thing is an emotive thing. It's there's a whole load of emotions going on in here. Not only the feeling of danger with the virus and the environment, but also the feeling of helplessness that people have when they can't contribute. And so managing that's really kind of important. And I think Andy was saying that he's got teams set up or Zoom and things like that to keep in touch with his staff. These are all good things, and we do them too. But also, if you can keep them active in some form, is also good self-improvement and things. That's just a point I wanted to make. Do you remember what you were going to say now, Peter?
Yeah, I do. Thank you. I'll start with that. The splitting between days, 2 days, 3 days, 3 days, 2 days, I think is an excellent way forward personally. It also minimises the amount of people coming into that space from day to day. However, there's a lot of institutes at the moment looking at half days. So you've got one team in the morning and another team in the afternoon. I'm not sure how that works for science research particularly, and which is why I'm hoping that we will follow a model where the leads of each lab can help contribute and say that's what works best for their area. That'll be interesting to see how successful some institutes where they're recommending afternoon and morning. I think I saw someone else mention morning and afternoon shifts. And again, for someone with childcare needs, that doesn't always suit them either. Certainly extending hours. And just coming on to, I'll come to you in a second, Alfonso. Just coming on to the other point, you using Microsoft Teams, we have Zoom meetings at least twice a week, probably 3 or 4 times a week we're meeting as a team, even now when we're not in. But what I'm going to encourage is to have virtual coffee breaks when they're in the lab. So there is a dialogue, or at the end of the day, that they can almost do a handover from the one team to the other team, explaining what they've done, what's coming in, and have a handover so they know that they've got interaction, and to make sure there's no walls. And I should mention in the chat that someone mentioned, those at home can be doing analysis as well. And of course, coming through. So Alfonso.
Yep. So one of the, when we are talking about core facilities and research, we cannot forget that we have core facilities with just one person. In fact, in Ireland, all of the core facilities is one person. So in this case, we all have small children at home. Some of us, we are going public transport, and we have to take all the responsibilities in just one go, and we have limited time. So we cannot cover all the users. So education, 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 and rely on the users that they will do what they have to do. So what I'm thinking is provide access to very good users, and they will be the ones troubleshooting the other ones, or just the more advanced are the ones that they will go at the beginning in phase 2, 3, or 4. Newer users, they have to give the samples to the more experienced users. I think that's the way we will manage. So I'm asking for any extra ideas in this area of one single core facility.
Yeah, it'd be good to hear some opinions or some ideas on that area. That's obviously very challenging, but if you've got the childcare, then it's a real issue, isn't it? Because you can't really restrict it with anyone, and you're going to be needed in the lab a lot of the time.
And at the moment, the childminders are not coming. There's no crash open. Thinking on the schools, you have to pick them up afterwards. So if you have your partner, and your partner is also working, you have to make rotas as well.
Maybe this is where, as Andy mentioned earlier, some labs may have, he thought the super users in labs that sort of delegated. The departments have to pull together right now, and maybe it's a case that they have to draft in some super users to help the core facilities and allocate some of their time. So actually, if they're only allowed in 50% of the time, maybe the other 50% they are in, but are helping the core facility rather than in their own lab. It's great for their CV. So that'd be a good aspect to it. Good to help other users, get them better known, so there can be real benefits to them as well at that point. Andy, Philby.
Thank you.
Yeah, I just want to say, Pete, that's a really good point. Bringing in kind of extra help. I mean, our animal unit at Newcastle would not have survived if the person who runs that, the director, hadn't been able to recruit other people to come in and do cage cleaning. In fact, her model of running has been incredible. It's something that we're actually looking at as well. It's very incredible the way because she obviously has been running the whole time. So I agree. Bringing other people in with skills could be, ringers, let's call it. Yeah.
These staff are being paid to be at university when they're having to work from home, and if they can't work productively at home, most will actually be very pleased to be able to still justify their salary. This is, those who have been asked to work are probably feeling more comfortable than those who are unable to work or not given the work to do. It's quite an odd situation where both sides mentally can be quite distressing and disconcerting.
I think we're in quite a good position, aren't we, in the sort of labs that we're in that we do have things that we can do from home. I think that there a lot of educational stuff, a lot of resources. I think Andy mentioned in the chat there, it's opportunity to read papers, write grants, and so on. So there's stuff to be done, but you do have to take into account, as Pete was saying, people's feelings about that, and there is that feeling of guilt, I think, if you're not at work the whole time.
We also need to factor in that if you have your team set for a week and then suddenly one of them has symptoms or somebody in their family does, and they suddenly have to have a week, possibly 2 weeks off work. I mean, hopefully the testing will be there for us to know whether it pretty quickly, if it is COVID, that we're then going to have, other people are going to have to come in last minute, or experiments may have to be cancelled.
Yeah. Eric wants to say something. Before I bring you in, Eric, can I just throw something out there that some people may be able to answer? If you are running these two teams together, are you going to run them completely separately, or are you going to be mixing them occasionally?
I think you have to. I think the instruction, certainly in the UK, is to keep them independent to each other. If you start mixing them, then there's not much point doing teams. It is to try and make their own family groups almost within the lab. I'm sure that's what the government guidelines is suggesting. Eric?
Hey, I won't use my video, but just something. So my workplace is team A, team B, 2 operators per week on a weekly rotation. Something that actually happened to us was one of the researchers who came in to run a SOT was in close contact in a way. So they came in 1 week on a Wednesday, and then the following week on a Wednesday again. And then in the third week on a Tuesday, they called up and said they had to go for a COVID-19 test and tell all four of us to go to sort of like isolate until the results was out. So we were like, oh shit. We had a chat with WHS and our the people and culture, or what we call HR. And they said, oh, well, the first two guys, the two from the first week can come back in because that's after 14 days. You have not interacted with the person for 14 days. So that's fine if you come in on a 15 days. So we didn't actually have to break our operations. So that was pretty all right. That being said, in the second question that Derek was asking about, team A, team B's, whether we mix or keep totally separated. We had a little bit of mix currently, but we had a chat with the business managers and the leadership teams and stuff like that, and they were saying with the increase, starting with Australia, we're starting to increase in workload. That is going to change. That means we have to stick with full separated team A and team B. So that's going to be a bit of a challenge. At the moment, we are floating the idea of bringing in, say, RAs in research teams that cannot work from home to fulfill their work. And if they're willing to come in, they could potentially be trained up to help with SOT operations so that we can increase capacity even with current 2 operators.
I think that all sounds very, very sensible. And kind of echoes quite nicely what's been said and backs it up. Thank you very much for that, Eric. Derek, to answer your question, if you're looking at the chat, he's actually these teams seem to be fairly separated, which does make sense. I'm going to throw something out there though, which your question raises nicely. If we work a 3-day, 2-day type shift, and the department teams are working 1 day on, 1 day off, actually our teams are being exposed to lots more people. Whereas if we follow the same pattern as a department pattern, that would also minimize the chance of spread from one team to another. That's something I hadn't really contemplated until just. So if we do have to do an 8 till 2 and a 2 till 8 shift, we probably have to fit in with that so that our staff are not mixing with the different teams outside of the core facility. I don't know if anyone's thought about that or how that would be managed. Is anyone working a facility with shift patterns that are different to what their department is requesting? I guess the UK, most of us don't know because most of us aren't in that position yet. But there's a lot, there's 21 other countries out there that must be doing this at some point.
Yeah, I mean, Karen raised a point there in the chat, Pete, that there's also the issue of public transport. And not everybody has the luxury of being able to drive to work or get the bike there, especially in London. Obviously, they have to rely on the public transport, and that's a bit of a who knows what it is at the moment.
Yeah, and yeah, if you live some distance away, unfortunately, the bike doesn't work. Unfortunately, for some.
Anyway, Chris, let's bring Chris in. Raise your hand.
Hi. I think it's going to be really important for people to be able to remotely control their instruments, to be able to use TeamViewer and the like. Because I know a lot of IT departments don't like it, but there are ways around the fact it's Windows 7. You can have TeamViewer whilst also disabling internet access, for example. So right now I'm in quarantine. So I'm the only lab member, and I can't be in the lab for 2 weeks, and the lab's reopening potentially very soon. So all of my assistance is going to be remote. And if people become ill later on in bigger labs, or if you're running a small lab and you become ill, you're going to have to do the same thing. So I think it's important to try and push your IT departments to allow some sort of remote working.
Yeah, that's a very good point. We'll come back onto that staff user interaction, but I totally agree. If one member of staff is in the lab, even if you've got more, if you've got 2 teams, it could be because we might not be able to cover the same breadth of equipment if we sell sorting at the same time, that we can, if we can get someone external to remote desktop in to help optimize the settings and desktop share at that point. That is an excellent suggestion. I'd just like to go back, something I picked up much earlier in the list, a lovely sentence or paragraph from Lola, actually. And actually, the things we can add to our users, so it's kind of moving just off our own staff, but our own staff can do drop-in surgeries, asking users to send them problems and questions. We asked, all my staff have been actually pretty flat out in all cases, attending webinars, upskilling themselves. So when they're out of the office, they're in there, they're doing webinars. We're having regular meetings. And as Lola points out, we've been very flexible in those meetings and had enough that if someone can't make it, they can't make it. It's not a big problem. So it's not compulsory at that point, but actually everyone does try to attend because we do have to accommodate those different shifts when they are home tutoring as well.
It's also useful to communicate, I think, to your end users that because people aren't physically there, that they're not out of the loop, and that they can be used for remote training and questions about analysis and so on.
I think Christian's just brought up a really nice point as well of having early shifts and late shifts. It means we can extend the number of hours that our labs can be used for, not just our labs, but the research labs as a whole. And that's possibly why they're looking at morning, afternoon, because it spreads the amount of working hours available. Karen, you have your hand up. Thank you.
So I might just echo what Pete said, but I got distracted by children in the house, so apologies if it's a repeat. So with staff, what we did is we had a user group meeting for the whole department and for anybody in the university that wanted to attend. And that was really good for the staff to show to the rest of the department and our user base that we were still working, and to encourage them to interact with us to help with data analysis. And that was really good for our user base to see that we were still there for their work, analysis and assistance. But also good for the team to show that we could still collectively come together and make impact. And I think further down the way, we might well repeat that again after lockdown, because we're not all going to be able to be in the same room and have a user group meeting, hear what the users have been doing with the equipment and new research or new applications they want to do. So that might be a way to help share the workload, because you can discuss what needs doing, but also keep the morale up and keep the team and the user base together, and to bounce ideas off each other, and to keep the flow cytometry alive within the department.
Yeah.
Sorry if that was a repeat.
No, that was not a repeat, Karen. Thank you. I hadn't mentioned the user group meetings themselves. The other thing we've had our team doing is using Google Chat a lot throughout the day. So whatever they're looking at, any problems, any topics, any webinars, they're talking about it to the rest of the team. So they're remaining engaged with everyone's activities and thinking, well, that sounds good. I'm going to have a look, or I've got a problem. And then they will Zoom in together, help solve the problem together. So they're working remotely really effectively. It's worked really quite nicely because we haven't had users distracting us every few hours, minutes. So it's got its benefits as well. I think we've been probably almost as productive, but that's going to become more and more difficult as results start to dry up and all the analysis is done at that point.
Yeah, we're sort of moving on, aren't we, there to more interactions with the users.
Yeah, so if we go back then to that staff rotations, this is a real concern. It'd be nice to know how concerned people are going to be about it. We may just think about children because that's a big one, but think about it on a bigger scale in the poll. But public transport, it's a huge risk. We've minimised all the risk in our lab. We made everything perfect, and then we got someone walking in that's just caught it. And they won't show positive anything because it'll be. It's not easy, is it? It's going to be a real challenge to do that. So I think that will move into how much work can be done remotely. But I think we should probably move on to.
Well, Andy Riddell, you've got your hand up again.
It wasn't on mine. It is now.
He is, yeah. He keeps putting it down.
I was just going to add to the mitigation of risk. All you can do is what you have in your lab and no more. Everything outside is everything outside. And if public transport and other things are to bring your staff in, then, I mean, what I'd tell mine is to come in off-peak hours and make sure the train isn't overloaded. If the train is overloaded, don't get on it, and just come in when you can come in. And I mean, just follow sort of self-awareness, be a bit more aware of your environment and stuff. But there's nothing you can do about it. Outside is outside.
Yeah, that's absolutely, Andy. It's just a case of how, if we've got people who can walk and cycling to work, do they get priority working in the lab? Will they feel aggrieved because they are able and the others have to use public transport? It's managing those tensions that can occur. That
just pay them more.
Go on, Andy.
Just pay them more. Yeah, they do need some certain amount of reward here. There should be something set aside for them, the ones that can make it. I mean, most people genuinely want to do something. I mean, without a doubt, that's true. And they get frustrated and emotional about not doing stuff. So, you know, we've got to take some care over our staff. And I think if we can reward them, either payments or with extra holidays or you know, some sort of recognition of the work that they've done, it'll be a good thing.
Yeah, and Cameron Underwood's just made a lovely point. It is true. We are going to shops and all sorts of other things where we have risk. There is no such thing as no risk in this scenario. It's just trying to balance that and manage expectations. We do have to be careful at-risk groups, and certainly in the UK, if they are deemed an at-risk group, they will probably not be allowed in anyway in the UK. Maybe different elsewhere, but children will not be classed as a high priority group in any case. And managing that with staff could be interesting. So I think we'll call the poll actually now, because the next topic is going to be a lot larger to discuss. So this is, should staff with small children be expected to work in the facility in person if there is no school or daycare available? This is a quite fairly clean-cut answer that's coming through, which we'll share in a moment. Just give it 5 more seconds. Quickly get your votes in. Cool, look at that little flurry of votes at the end. Okay, calling an end to the poll. Going. God, I feel like an auctioneer. So 67%, do you know, concentrate on only 5% think that they should still come into work and be expected, which is a very low percentage. What I should then challenge, so who's going to do the work, and is it right that you have to do the work because your children are older or you don't have children? And that's the tensions that I think can be seen in the labs. I think that's a really nice example of how people's feelings are about this. And it's a time where we do have to pull together and help those who have got other commitments that we don't have. That's a very nice, clean cut result, I think, even though 27% unsure. But thank you for doing that poll. I shall now hand over to Derek, because this is a really hot topic.
Well, I think there's obviously some of these things have come up already. We've seen the overlap between the commitments we have to our staff, but also the commitments we have to our users. And part of that, of course, is training and helping them, whether it's acute troubleshooting or whether it's starting from scratch. And we've already seen, I think in the chat, a lot of people have already started talking about this, and a lot of people have stopped all training completely, which obviously may be something that's going to work in the short term, but we don't know how long we're in this for, do we? So it's not going to be something that's not going to go away. It's not going to go away. So we're going to have to maybe think of other ways of doing the training. Now, it is possible to do remote training. I mean, there are lots of people on this call today that are very experienced in remote training, but there are still occasions when you need to have that face-to-face interaction. So how are we going to deal with that over the coming months or even years? Do we, sorry, say the last little bit, do we expect to change the way that we're working? I think Andy touched on it earlier, doing more full service work, people not just dropping off their samples, but dropping off fixed things that we then do the staining of, which would then have more implications on our staff time. Sorry, Pete, now you can come in and say something.
Anne Wilson's up. Go on, go.
Okay, Anne.
This is something that we've been discussing in Switzerland amongst all of the core facilities actually for the last 3 or 4 weeks, because obviously we're all facing the same thing. So currently we're not doing any trainings 2 weeks in, but we are gearing up to the fact that we will have to do trainings in the future. So Derek has some good points. We offer already a full service at an increased price, of course. So our first option currently that the users have is that they send someone else from the lab to run the samples of the untrained user, but this is not always possible. Second option is they pay 4 times as much per hour to have us run the samples for them, but I pointed out to the PIs that we'll do it a lot faster than any of the users themselves. And that goes for fixed or unfixed samples. And then the third option is what we're currently setting up to try and do, and that is to set up training remotely. We are making videos. I had my guys in the lab Wednesday night this week making a video on how to use a Fortessa, the basics, the sheath fluid, the waste tank, how to put the sample on, how to turn it on, how to turn it off. We also have had access, because I had to do a training course, to some videos that we found online on YouTube. I think it was YouTube or a webinar where they actually have FACSDiva software. There's a section on FACSDiva software, which is better than anything BD has. So we're currently looking for all the different options that we can use to do it all remotely. We've had TeamViewer installed on the analyzers. We use this for remote troubleshooting already, but we're adding it now for training, and we plan to work with some audio and video link along with TeamViewer. So you remotely, or me remotely sitting in my office or my home, can run the computer on the machine. And I think this will work pretty well to guide the users through once they've done some YouTube videos and training, because there's a lot of information available on the web at the moment for that.
There's some interesting discussion in the chat already about the use of phones to do remote troubleshooting.
We use WhatsApp phone. WhatsApp video is fantastic.
And do you want everybody to have your mobile number? I have before now had a user call me at midnight to ask me what channel Daffy is on.
It's channel 4.
For the last 5 years, we have posted our private mobile phones with up to, saying up to 10 o'clock at night or something. Our users rarely use it, and when they use it, they don't abuse it. But in this situation, we figured for training, this is probably a really good way of doing it. I mean, some people have institutional phones, of course. That gets around your private number. But yeah, that is a downside.
Yeah, I think there's at York at the moment, you have to log in and log out of the department at the moment for health and safety reasons, because there's limited numbers in the department, and your phones are needed therefore to also call for backup and help. I think phones are actually essential to have on us now. If you've got them, I think you should be allowed to use them. You should be able to have access to them on your person. They're very beneficial. And I think being able to call for help is much better than having to leave the room to ask for help. If they can just call for it, it's much more efficient.
Yeah.
If they can get a signal.
Yeah, I think there's been a lot of debate, isn't there, around the use of personal phones in the lab. But I think we may be fighting a losing battle there, because they are useful, and people need them. You do need to be contacted.
Some Cat 2 facilities won't allow phone in there, so that would therefore is a decision from the Institute's health and safety whether or not they allow a phone in their facility.
And yet you're allowed the same shoes in and out the same lab. Mute argument. I'm intrigued by Andy Philby's glasses.
Yeah, I mean, they're quite useful. I've never actually seen Andy's glasses, but I know we've spoken about them a lot, that they are quite useful, especially if you've got remote site, even in normal operation, if you have remote sites, no pun intended.
Yeah, just looking at Eric's comment about not letting users use the sorters to minimize downtime. What we can still do though is turn instruments on, get them up and running, and leave the user looking after their sample to and come out and help others. So they can monitor the sample and we can go back and clean up at the end. So there can be hybrid models that may work in a sort lab.
I think somebody made the point as well a little bit higher up that, you know, you have to be careful in the longer term that you don't sort of lose business if people think of other ways of doing things. So you have to facilitate them being able to do what they want. And maybe people who don't train people to sort, that may be something you might want to consider.
A really nice example came from the light microscopy meeting, that someone who's training them or optimizing their experiments, the person may have to leave the room for the other person to go in, do some optimization, walk out, because it's a very small room, and the other person go back in. But obviously you can't leave 10 minutes between each visit. That would become too cumbersome. So I think desktop sharing is going to be critical in many cases for us to help users once they are up from the basics of how to change your fluidics, how to put your SIP tube on, and so forth. But I think it's Alison, there are Cat1 laser pointers that can be relatively safely used to point out bits, so we can guide them through it from a distance if the lab is big enough, and talk them through what to do. And remote mice that we can operate at a distance as well.
Thelma?
Hi. Just a question regarding trainings, because of course this is tricky, but I think some companies already have very nice videos online that we can use them, right? I think even for the Diva software, BD has something that it helps. The SciTech for the Aurora already also has something. And it's already like, instead of being 2 hours with them explaining, this is a tank, this is, you need to empty, you need to refill. These videos already help. And I think we are a big community. If all of us are doing the same videos, it's kind of a waste of our time, right? Also, the company should help us with this. And this is something that's, I think they should really help us with this.
Yeah, I mean, it doesn't necessarily have to be the companies, does it? We are a community, so we could have some community curated resources that help with the pre-learning, so that people have some idea before they come to the hands-on, and that would cut down the one-to-one training that we have to do.
Exactly. And I think, like what Peter said, if we are far away and they are just doing it for the first time and we are overlooking it, then it works, right? But I think these videos, and to have it available for everybody, would really be beneficial for sure. Like the sense.
It would be interesting to know what the guidance are when you cannot be socially distanced and you have to work in close proximity, if there's different PPE requirements at that point and how we conform. And I haven't seen anything on that, because that may enable us to do one-to-one training. But again, as Karen, I think, mentioned earlier, face masks can be very difficult to use if you've got someone who's hard of hearing.
Yeah, I think somebody mentioned maybe in the light microscopy one that obviously not everybody has English as their first language. You're wearing a mask. That makes it even harder to do it. But I think, you know, whatever happens going forward, this shows us that we may have to think about training in different ways.
I think Thelma came up with a good idea. There's a lot of us who have made YouTubes of guiding a user how to take a tank on, empty it, fill it, clip it back on. You know, actually we have that ourselves. So can we not find a central repository to put all those YouTube clips clearly titled? I know that I'll find the RMS website in a minute that has a load of useful webinar resources, and we could easily just tap into that and just push them on there, just link to them and share those between us all. I think it'd be a great community thing to do.
Oh, Jessica's back saying that the ISAC have a YouTube channel as well, so they're putting content on there with if the practicals, and of course there's also this is a good reminder that ISAC has the CytoU, so the webinars that are available and tutorials from Cyto and also from on different topics, and they're free for everybody at the current time as well. So that's a good resource for your staff and also your users.
Yeah, the RMS one is very light microscopy or biological microscopy orientated, but it's still useful. I will put it on the link now.
Yep. Karen?
So I think with the flow cytometry and the way the analyzers are now, at least we do have the advantage that if the data has been collected on scale and appropriately, if the compensation is incorrect, other than for sorting when it's obviously then gone wrong and it's really wrong, we can let the user have a go at setting up the compensation, and then they can walk away and we can go back to the machine or look at it remotely and maybe tell them what they've done wrong or advise them how to do better. So in some ways we have that advantage with the modern analyzers, and that can be, you know, useful. You can have a dialogue after they've had a go and then correct them where they might have gone wrong, and that'll be a real benefit. And anybody that's got avalanche photodiodes as well with the settings, you know, if they have used it at different gains, it matters even less, but not everybody has that. So I think we're in a fairly good place with the remote videos that we've got, as well as the way the technology works, to, you know, make this not insurmountable, but eventually it will need some one-to-one. And if we're going into bars and restaurants and supermarkets, then we can probably cope with some limited, you know, not close quarters encounters with our users, but with, you know, sensible, limited, you know, interactions within close proximity to correct mistakes that can't be done remotely. So I think training, you know, should be able to proceed in the fairly near future with caution and all the tools and resources available. So I think getting our heads together and putting together a pool of resources of videos that are instrument specific and software specific would be great, and a really useful resource to help implement this. So yeah, that'd be really good.
Yeah, I think, you know, caution is the watchword there, isn't it, Karen? Because a lot of the interactions we have with users are relatively, I was going to say casual. You know, you're not with them for a long time if you're troubleshooting an acute problem. But with training, you tend to be there for a lot longer. So we need to just make sure that we're managing the risks appropriately.
I also wonder whether there'll be a reduction in the amount of training that's needed, that maybe we're not going to be having the summer students, and there will be postdocs not starting straight away. So we may have a few months to get this sorted for, you know, maybe the autumn when we might have new members of staff and students.
Pete?
Yeah, just to go back to Christian's question about the mouse. Yes, just a Bluetooth mouse in at the side, and then you can work from the back looking at a distance. The only problem is some of the screen resolutions might not be quite so easy where you're going, but if you can get people in the right place, virtual desktoping will be best, but we've got to be able to use it on our Windows 7 or even Windows XP in some cases, which could be a challenge because they're not networked necessarily.
I mean, there are all sorts of issues, aren't there, with some of the desktop sharing programs, particularly TeamViewer. I think that has quite a big cost associated with it if you're not using it for personal use. It doesn't work necessarily with the Windows 7 environment.
I believe Zoom can work on Windows 7, I think. But again, you've got to have it on the network to enable that. Something companies could really do for us, vendors, is to enable us to upgrade at a much lower cost, because there's real financial constraints now. But to enable us to keep using these, we need some help and support.
Exactly, and all Windows 7's machines are off the network, so that gets rid of that problem.
I believe you can use if you can get it to a Windows 10 PC and route it through, you can then get to the outside network via that PC and still desktop share your Windows 7. It enables a virus-free route to get out. That may be possible, but it's not something, if you haven't got it set up, it's going to be hard to set up now in the short term. It may be that we're only in this position for a month or two, and then we're essentially working under almost unrestricted, just very careful monitoring. I do think it's important to deliver as best we can in these couple of months, though, because when university finances are hard, they are going to be looking for job cuts, and I don't want it to be any of us. So we have to show our worth and our need.
Yeah. That's a great thought to put into people's minds at 20 to 5 on a Friday evening, Pete. Telma.
Exactly, that's a great way to finish. Anyway, I'm just because we are already on for a while, and before we had a regulation, one person in the room, and now I have two. And at the beginning it was nice because we could be, especially for the sorters, I need to be with the user. Sometimes it's tricky to start one. We have two persons, but still the 2-meter rules, and it's difficult to make them follow this. That's something to keep in mind. when you have 2 persons in the room, some users, they don't know what's 2 meters. So we always need to say, just back off. So that was also hard, but yeah.
But you can hopefully you can put tape down in your room.
Yeah, but you know, then they don't see, and then they want to do something, and then you see it, and they are right here. Like, come on, you're not supposed to be close to me, and that has been hard. I think this has been the hardest thing so far.
If there are Americans doing that, tell them it's a yard or 2 yards.
Exactly.
Karen, you want to come back?
Yeah, although this is staff user interactions, I was just curious as to the people that are working and have had engineers in while they're working. Have they had any issues with user engineer interactions, or any delays, constraints, or difficulties associated with that type of contact?
Yeah, that's a good point, Karen, actually. So that sort of didn't quite come up earlier, did it, when we were talking about physical distancing? Obviously, the engineers do have to get in, they have to comply with everything. It's no different. So how do we do it? How are people doing it?
Yeah,
those of you who are working.
Yeah, that'd be really good to hear.
Must be good to hear from somebody.
I can try and volunteer somebody from my lab if they'd like to say how they did it with their engineers who've been in. I wasn't in there, so somebody put their hand up.
Hi, Marie. You're on.
It's when you have split screens, finding where the microphone button is really hard, isn't it?
Yeah.
Marie?
You got your hand up, Marie.
Yeah, sorry, I'm new for Zoom. Can you hear? Okay.
Yeah, we can hear you now.
Yeah, okay, sorry. Yeah, no, we've just offered them masks and lab coats, tried to maintain the distance. It hasn't been a problem, really. And we've gotten very fast service. So this was one of the benefits of being open when most things are not. I think we had 4 different service engineers come for scattered different machines, and it hasn't been a problem. It really worked well.
We had a risk assessment for them to sign. So we've got a new risk assessment for all our users who are using the facility, and I've also sent it to engineers as well.
Oh, that's a good idea. Yeah.
Richard, you had your hand up as well. Hi, we can see you.
Hello, can you see me now?
We can see you and hear you.
In the lab, running samples most of the day today. We've had a few engineers in, mostly Fluidigm actually, on several instruments, and it's been fine. As far as I know, pretty much all the engineers have been available. None of the companies have said they haven't had people available. And we've had quite a few people plan to do PM visits while it's quiet. We haven't had those in because those machines haven't been important or being used. So we'll plan to do those when we're back again.
That's a good point. Do you think we should do more full service work, or should we carry on as we are? But Richard, I know your lab space. I know even your office space becomes impinged with the lab space as well. So how?
I mean, yes. Well, my team are now out of the actual lab. They've got office space somewhere else, but my lab's still in here, and so I'll be walking behind people's chairs to get into my space. So we're going to have to really we're figuring out one per bay, and then going by institute guidelines on either masks or whatever.
Thank you. I just listening to the conversation, I think most are thinking that it's still better for users to be using the cytometers themselves and not doing more full service work than we were previously doing. Which might mean if we do have new groups, we may have to do it for the new groups that are coming in, or we're going to have to train the new groups. I think short term, I think it's going to be a pragmatic and what best suits, isn't it, at the time?
Yeah, I mean, also we've heard a couple of people, haven't we, talk about having the super user idea, which is something I think a lot of people have been resistant to in the past, but maybe this is, again, it's an interim measure that would help us. Andy, you have to say something?
Yeah, I guess I just want to say again about the challenge of being in a university. I mean, we're getting a lot of discussions about, the students and what the students need. And, a lot of the undergraduate projects did come to us and, they weren't that advanced. They were quite simple. But, the idea that maybe there will be no more lab-based projects for a very long time for undergraduates. Maybe it will have to be in silico analyzing data and that kind of thing. But it just worries me to think, if we do stop completely having interactions with them, then we're going to lose out, and we won't be fulfilling our educational requirements that we have for our different graduate courses and postgraduates. And and again, this idea of a knock-on effect, we may have a medium-term problem, a long-term problem. Again, as we said, other people may find other solutions for doing the work and not coming to us. I mean, it's not always going to be the case that you can find an alternative to flow, but it could be a problem. It could be a real problem. So we have to be creative, I think, safe and creative.
Yeah, I think a lot of people have said in the chat that their institutes are no longer accepting any summer students this year, in common with us as well, I think, and hopefully that's just this year. But if it went on, that would be a problem.
I thought Sarah Bennett just asked a very interesting question of, when engineers visit, who cleans it? Now, I think we probably have a responsibility to clean it before them, but they also need to be taught our local rules and have to comply to that as well. So I think of both. I can't spell should when I'm writing fast or slow. It will be interesting, won't it? Because I think we're fortunate. We may actually have more staff back than some of the labs will have back themselves. And because the labs are only at half capacity, our user base will only be at half capacity. We may be able to cope without too much hindrance. I'm ever the optimist. Much better than my last comments, Derek, on the last thing on a Friday. Much more cheerful and upbeat.
Yes, that's right. Well done. Well recovered. Richard, again.
On that bit of returning to work, our institute's planning sort of one person per research group. So it's probably only 20, 25 researchers, but each one could probably access every single bit of every single core. So I suspect it's going to be the case that all of our services, all the flow, the microscopy, the genomics, the histo, all of their capabilities are going to be needed, even though there's only 20% of the researchers back in the building, or 5%. So it's probably going to hit us disproportionately to the research groups.
Yeah, absolutely. I say we can probably have more staff in than the others to support that.
Yeah.
Yeah, but as you said, Pete, I mean, that does mean that you have to have a little bit extra time to deal with that.
Yeah.
At least in the short term.
Which would be very good if it was the case, because it shows the importance of the core labs and our staff.
Let's spin it the right way, yeah.
Gosh, too many chatters going on quickly.
Yeah, can't keep up with the chat.
No. Ah. Yeah, it does. If that 20% of staff coming in are all at bench researchers, that's like 50% of our user base, let alone anything else, not even just the fact that it's on 50%. And it will be those who are actively on the bench that will be coming in, not those who want to sit in their office all day, because they don't need to do that. They can do that from home in most cases. Not all.
No, but that's a good point. Yeah, people who are working from home will stay working from home.
Yeah, so actually, Lisa's got here that they're booking instruments block booking other instruments in the room at the same time. It could be a case that we look at the high priority instruments, the ones that are most used or most needed, and only leave those ones available until the last minute, and then the other ones can be used. So that the most important instruments, the most capable ones, get users on those, and essentially we have to kind of mothball maybe one or two other instruments. It could be a lot easier to manage that way.
Alfonso, you're back in the room.
wait, now here I am again, because I'm mute. So I agree with you, Peter. In fact, we have several instruments. You know the facility. For instance, we have provided access just to 2 of the instruments because we are sharing part of the lab as well with another facilities. We have MTA or Viacore or Luminex. So we just limited to 2 instruments because also if they need some of my time to be online and help them remotely, I have another commitments as well. So I cannot help with all the instruments at the same time because I'm not an octopus and I have a couple of computers home, not too many, not as many as instruments in the lab. So definitely blocking some of the instruments, that's good on one side. On the other side, that will help us. I was looking at one of the comments from Bergen that they have problems with the engineers to come in. So if I have a breakdown, I can have another instrument as a backup instrument.
So just a thought, there's a lot of teaching labs in many institutes, not all, obviously. And that teaching lab space will not be used for a little while, at least until October.
Mm-hmm.
And there's clear space that we could move some of our equipment over to. The Luminex could be moved pretty easily across to give access to it. And just thinking about Rachel's question as well, a lot of the lecture rooms may not be used, or the small meeting rooms. So we could repurpose those for office space, not just for core staff, but staff in general, to give bigger spaces. So I'm not thinking hot desks here in some cases, but giving people space that it wasn't typically theirs. So it's not causing the crossing over of teams and stuff, but there is a lot of space that may not be used during a partial opening that we could spill out into.
I agree with you. In fact, we are moving to Luminex, to System Biology Ireland, which is another building, and they are the top users. So we are bringing that to their lab, this instrument. So it's a question of that. We can also use the Acuity and move it to the other side, but if the Acuity breaks down, I have the second one as a backup. So I prefer to use it that way, because also sending instrumentation, or as I point out, bringing an engineer from another country could be an issue. Mm-hmm. So I prefer to have them as backup in the facility.
And the other thing to obviously think about, not just teaching labs, but there may be other labs, core facility labs that have equipment that is a lot less used than some of ours, or vice versa. So we might give space to a system that is much more used, a qPCR, for example. But then there may be other labs that is not very often used, and we may be able to move some of our equipment into there. So I think we need to look at it outside our little world of our flow labs and look at the bigger picture of where we can move or how we can help as well.
Yep, good point, Peter.
And Karen, I don't know why you're asking about coffee and lunch breaks. You shouldn't be. You shouldn't be getting those. Just work harder.
I'll just wait for the comments to flood in there, Pete.
Something controversial at the end.
Yeah, Alex, a hand up.
So hi.
Hi.
How are you? So let me just give an input in the other side, because we give some support to the customers, to the core facilities in Portugal, because I'm from Portugal. So one of the main difficulties that we are facing is that some institutes have a policy rule of not allowing anyone to get in except in certain circumstances. So in most of the cases, we are not allowed even for a maintenance to be done, and they are postponing most of those maintenance. And once the instrument breaks down, there is a need of an authorization to get inside to make the repair, and they almost tell us to make the repair as quickly as possible. So sometimes it's more difficult to get inside and go around some of the policies of the institutes. And my key point is moving some of the big instruments might be spreading the problem and not concentrating and putting the people aware of some difficulties. And moving to those places, to other rooms, it will be even tougher to control the usage, and not only to have access as a remote access, or even for the staff to be inside to give all the support to them. So one of the things that it would be crucial also is to talk with the companies to show the needs that you have, but also to understand that sometimes each lab has their own will, but then the board of director of the institute just locks down the door, so we cannot get in and help. Okay, so that's another point of view. And thank you.
Thanks, Alex.
Yeah, very good points made, and comes back to the point that we can only try and influence, we still have to go with local rules. Love the picture, Alex, by the way, as your background.
Alfonso, another point.
I thought that nobody from any company was here, so if Alex, you can answer this question, or you can move it inside, because I've seen that BD is not allowing remote access to the instruments when they have this remote possibility a long time ago. I remember when we started to do remote sorting, they were the first one embracing the technology. But now it's no longer permitted because it's coming something related to service contract. I was not aware of that. Do you think that BD could be taking off that problem and allowing companies to go for remote sorting just to provide access to the instruments? Can you move it inside?
I can tell you, what I can tell you is a simple thing. You know as well, and in Portugal we do things a little bit different, and they work, and we go around. BD is a big company. What I can tell you is, I see movements inside BD to put some of this system, the link system that they have internally, and I can talk with some people, and at least I know that we pushed for Spain and Portugal at least to have this, at least for the people that have service contracts, to include this free of charge during this period, and we are trying to extend. The problem, and I tell you in a minute what right now it's possible, is that, for example, when you go to this, the problem is that you can only give access, remote access right now with this platform, what they say a surety link. Okay, this is the name of the product that they have, to Canto, Aria, Celesta, Fortezas, Symphony. These are the only instruments that are covered. So instruments like calibers or any other older instruments would not be included in this, but at least in your areas and the rest, it's possible. I know there is a big pressure on everybody on this, and this is something that we are pushing at least, and I know that at least Portugal and Spain are pushing a lot to have this, and I think this has been discussed and will be spread at least in most of the countries. I don't know if Switzerland, because I don't know if they like Miguel a lot, but but I think everybody will be included in this. Okay, and I'm just a small fish here near the ocean, but I'll be pushing for everybody on this. No problem.
Good. Thank you very much, Alex, again.
Yeah, thank you.
Okay, we should probably move on. We've got a couple of slides left, and we've got a couple of minutes left. We just wanted to do one poll in this last session.
Yeah, please stay on.
Yeah.
Don't disappear yet, because we've got something else coming up after, so please stay on. Give it another 10 seconds because they're drying up.
Yeah.
Yeah, go for it, Derek.
Yeah, interesting, because I think the way that I was reading the polls, I thought that wasn't quite as far in that direction as that poll seems to suggest. But obviously, a lot of people are thinking that we'll probably have to do both of those things. do what we normally do and also do some full service work for people.
Maybe only for the first month or two, for doing that for where they're not trained. Maybe just quicker and easier to do it for them than to have to do remote training. It may just be faster. At least they'll know what good results look like at the end of that. So there could be a benefit here if we're doing it for them. Unbiased and good data. Have we shared the results?
Okay. Oh, I don't know. I may have cut it off.
No, it's on the chat.
Did
Oh,
everyone
yeah.
see the
It's back
result?
now.
Yeah. Okay.
Yeah. Yeah, but we put that into the chat.
Brilliant.
Okay, so we're almost at the end of this. But I suppose, yeah, one of the questions that we did want to ask people is, well, Peter, you wanted to do this one, actually.
Yeah. So the other meetings, we're proposing a follow-up meeting for people to attend. But before we ask a survey, what would be really good is if you could, in the chat box, put down any items that you'd like to have covered that we haven't covered today that we could also bring to the next meeting. So I know someone's asked about external users, external access. Finance.
Yeah, finance, that's a big one. Yeah, we sort of avoided.
The other thing that I'd like to propose actually is a less rigid format that we've had today, because we've covered a lot. And I think in a couple of weeks' time, some of us will be going back into labs. Some of you will have been working in a lab for a couple of weeks and know what's worked, what hasn't, and move into the next phase. And others will just be considering it, because across the world, we're all in different places and different times. So what might be quite nice is maybe a 1 hour open forum every 2 weeks where people can drop in, propose questions, and we just chat around the questions that people are really wanting help with, and to have good ideas and suggestions of best working practices. By then, we might have people who know how to get Windows 7 and Zoom in the external network working efficiently. And we may be able to share tips and tricks around things like that. So we've got the sample handling, the follow-up experience, but think about the open forum as well. Is that a good idea? Just have it more free format. You know, we've got this chat, people can just ask questions and we can just run with those questions live as it is. We'd probably bring it slightly later start time, so it fit in with East Coast and West Coast to the States a bit better. So it's not really, really early over on the West Coast, to make it more sort of across Europe and into the Americas as well. Quite hard to get it down to Australia and the APAC regions. But again, there's no reason this has to be only run once. We could also shift time zones and run a similar forum for other areas. The RMS is an international society, and I'm sure would help us set that up if we deemed it necessarily. So please have a quick look through those chats. And I think, shall we go to the poll?
We have one very final poll.
Because if we have a meeting, we do want people to attend and make it useful, but it doesn't have to be mass numbers. If it's every other week, it could be, it's quite nice actually.
In fact, it might be better if there are smaller numbers.
Yeah, and you know, it'd be really nice. I think it's nice for all of us to see people, to interact, to have the dialogue, know we're not alone in our little wells of running core labs of cytometries. I think it'd be really neat.
Somebody's asking whether or not a forum would be good. I don't know whether that's easy to set up a...
Yeah, Adam, could you please try and set up a quick poll? So we've got the follow-up, but is a forum, or let's have a quick, is a forum the best way forward, or more like today? So free format forum or a very rigid topic driven forum? What would be best?
Is there space for both?
Yeah, end that poll. Share the results. So it's pretty conclusive that everybody would like some form of follow-up, because I did say that this would last 3 hours, and you know, there will always be things that we won't be able to fit into the discussion.
Let's see if Adam can get that other poll to know which format might be most appreciated. There we go.
Well
Thank
done,
you very
Adam.
much, Adam. That was super speedy. Thank you. And thank you everyone for the thanks that are coming in.
While that is going on, we'll put that last slide. Yeah, as Pete was saying, thank you very much. If you have any suggestions that you think about later, you can email us, and I'll just put my email in the chat. I'm sure the others will do the same. If you think of anything later on, please get in touch, because we'll try and facilitate that. It does look like we will be following this up in the next few weeks.
Yeah, so please do email us. I think the open forum as well, I think there's clearly points like finance that would be on the agenda very early on that. Straight off. Okay, I think
thanks, Alfonso.
I'll end that and share results. So majority for a combination of both?
We just have to work out how we do that.
Well, I think if people send in their points that they would like to discuss in advance, we can have 2 lead topic points and then free format as well. It may need to be an hour and a half. So because we do need time to discuss, but we'll see how they go. We don't need a strict end time. You know, we can have 2 points, spend 20 minutes on each point, and then free format until it runs out. People can drop in, drop out. It's not like a conference where you feel guilty sneaking out at the back of the room. Yeah, well, thank you very much, everyone.
Yeah, thank you all. We'll finish there, and hopefully we'll see you soon.
Yeah, enjoy your weekends.
Thanks for coming.
Yeah, thank you.
Useful.
Bye, everybody.
Bye.
Stay safe.
Bye.
Thanks, Karen.
Bye.
I've never seen the room empty so fast out of all.
That was quick, wasn't it?
That's a Friday for you, isn't it?
Are you still recording?